and welcome to the Sleepy Medicine channel. You're in a quiet place now, where medicine slows down and the body remembers how to rest. Here we explore the gentle side of science, the processes that heal quietly, the systems that work in the background, asking only for calm. As you listen, there's nothing you need to do. Your body already knows what comes next. If these peaceful medical journeys help you, you're welcome to like or subscribe. It helps others find this space too, one resting mind at a time. For now, just let your breathing slow and allow your thoughts to soften. Let us begin a journey tonight, a slow, gentle voyage that will take us to two seemingly impossible destinations at once. Close your eyes for a moment, or let them rest softly on these words, and imagine yourself standing at the edge of understanding, where the infinitely small meets the incomprehensibly large. There is something deeply calming about this threshold, this place where human cells and distant galaxies whisper the same secrets in different languages. Tonight, we will descend into the landscape of a single human cell. We will make ourselves impossibly small, shrinking down past the level of what our eyes can see, past the resolution of ordinary microscopes, into a world that exists within you right now, at this very moment. Every word you read, every breath you take, is supported by trillions of these tiny universes, each one a complete world unto itself, bustling with activity, yet following rules so ancient and so reliable that they seem almost like meditation in motion. But here is where our journey becomes interesting, where it takes on a quality that might help your mind settle into that pleasant, drifting state before sleep. As we explore the inner world of the cell, we will occasionally glance outward to the stars, to the galaxies that wheel through space at distances our minds can barely fathom. And we will notice something remarkable, something that has fascinated scientists and philosophers alike. The patterns repeat. The architecture of the very small mirrors in strange and beautiful ways. The architecture of the very large. This is not to say that a cell is a galaxy or that a galaxy is a cell. That would be too simple, too literal. But there are symmetries, echoes, rhymes in the structure of reality that span from the microscopic to the cosmic. The same principles of organization, of energy flow, of information storage seem to apply whether we are looking at something a few micrometers across or something spanning hundreds of thousands of light years. Why does this matter for sleep, for rest, for the gentle quieting of an active mind? Because there's something profoundly soothing about patterns, about repetition, about the discovery that the universe, for all its vastness and complexity, follows certain reliable rules. When we see the same structures appearing at different scales, it suggests an underlying order, a fundamental reasonableness to existence that can help the worried mind relax its grip. The human brain loves patterns. It seeks them out, finds comfort in them, uses them to make sense of chaos. When you were a child, perhaps you found it easier to sleep after a bedtime story that followed a familiar structure. The hero sets out, faces challenges, returns home safely. The repetition, the predictability, created a sense of security. In much the same way, recognizing that the cell in your body follows architectural principles similar to those governing the grandest structures in space can create a similar sense of cosmic security, a feeling that you are part of something comprehensible, something that makes sense. A microorganism, something we will discuss later, contains within its single cell all the machinery necessary for life. It is, in a very real sense, a complete universe. It has boundaries. It has an interior space. It has regions of high activity and low activity. It has mechanisms for generating energy and for storing information. When we look at a galaxy through a telescope, we see something remarkably analogous. A boundary region where the galaxy fades into intergalactic space. An interior filled with stars and gas and dust. A central region of intense activity, mechanisms for generating energy through stellar fusion, and information encoded in the distribution and motion of celestial bodies. Tonight, we will take our time. There is no rush. Sleep will come when it comes. And until then, we will wander through these cellular landscapes together, occasionally lifting our gaze to the stars, noticing the ways in which nature seems to have used the same blueprint at radically different scales. We will learn about organelles, those specialized structures within cells, each with its own function, its own character, its own story to tell. The word organelle simply means little organ. And just as your body has organs like the heart and liver, each cell has its own tiny organs, performing specific tasks that keep the cell alive and functioning. Let your breathing slow as we prepare to shrink ourselves down. Feel yourself becoming smaller, smaller, until the fabric of your skin becomes a vast landscape, until a single cell looms before you like a planet hanging in space. We are about to pass through the outer boundary of this cellular world, about to enter a realm where proteins drift like clouds, where energy flows like rivers, where the instructions for building a human being are stored in elegant, twisted molecules. And as we journey inward, remember, everything we discover here, every structure we name and describe, is happening inside you right now. You are not just observing this world, you are made of it. Trillions of these cellular universes work in quiet harmony to create the person reading these words, the consciousness contemplating its own physical basis. There is something almost magical about this, something that deserves to be approached with wonder, with patience, with the kind of gentle attention we bring to the moments before sleep. So let us begin, slowly, carefully, with all the time in the world. Before we can truly appreciate the journey ahead, we must first make peace with the concept of scale. Scale is simply another word for size, for the dimension of things, but more importantly, it refers to the relationship between sizes. Understanding scale is like learning to read a map. Once you grasp it, entire worlds open up to your comprehension. Consider for a moment the width of a human hair. You have seen human hairs countless times, perhaps found them on your pillow, or watched them drift in a sunbeam. A human hair is approximately 70 to 100 micrometers wide. A micrometer, sometimes called a micrometer, is 1/1,000,000th of a meter, or 1,000th of a millimeter. It is a unit of measurement designed for things too small to see comfortably with the naked eye. Things that exist in that twilight zone between the visible and the invisible. Now consider a typical human cell. Most human cells are between 10 and 30 micrometers in diameter. This means that several human cells could fit side by side across the width of a single hair. The cells that make up your body, the fundamental units of your biological existence, are smaller than the smallest thing you can easily see without magnification. They exist in a realm that requires special tools to access. Microscopes that can magnify images hundreds or thousands of times. This is the scale at which we will be operating for most of our journey tonight. The scale of micrometers, the microscopic realm. It is a strange size to contemplate because our brains did not evolve to think naturally at this scale. Our ancestors needed to understand the size of predators and prey, the distance to water sources, the height of trees. They did not need to visualize things measured in millionths of a meter. So when we ask our minds to imagine the cellular world, we are asking them to do something slightly unnatural, which is perhaps why it can feel so dreamlike, so conducive to that floating sensation before sleep. But let us also consider the other end of our comparison, the cosmic scale. But let us also consider the other end of our comparison, the cosmic scale, the realm of galaxies. Our own galaxy, the Milky Way, is approximately 100,000 light years across. A light year is the distance that light, traveling at 300,000 kilometers per second, covers in 1 year. It is approximately 9.46 trillion kilometers, or 9.46 million kilometers if you prefer to think of it that way. Though such numbers quickly lose meaning for our human minds. Here is where something interesting happens to our understanding. The galaxy is so large that contemplating it creates a kind of mental vertigo, a feeling of falling upward into incomprehensibility. The cell is so small that imagining it creates a similar sensation of falling inward. Both scales defeat our ordinary perception. Both require us to let go of our everyday reference points and trust in metaphor, in comparison, in the tools of mathematics and imagery to guide us. Yet despite this vast difference in scale, approximately 43 orders of magnitude separate a cell from a galaxy, something remarkable emerges. Structural similarities. Organization principles that apply at one scale seem to reappear at the other. This is not magic, not mysticism, but rather a reflection of how physics and chemistry operate. Certain arrangements of matter are simply efficient, stable, or inevitable given the laws of nature. These arrangements show up again and again, like recurring themes in music. When we look at the scale between cells and galaxies, we find ourselves as observers. We humans exist at a scale roughly in the middle of this range, though not exactly at the midpoint. We are large enough to contain trillions of cells, complex enough to develop consciousness and wonder about our own components. We are small enough that the galaxy contains hundreds of billions of stars, each potentially hosting worlds we will never see. We exist at a privileged position for observation, able to look both inward with microscopes and outward with telescopes. There is something calming about recognizing our place in this hierarchy of scales. We are neither the smallest nor the largest thing in existence. We are not at the center of the universe in any spatial sense, but we are at a kind of perceptual center, able to observe and wonder about the full range of cosmic architecture. This perspective This recognition of our intermediate position can help quiet the ego, can help the mind release its anxious grip on the idea that we must be supremely important or supremely insignificant. We simply are at the scale we are, part of a universe that manifests pattern and structure at every level. As your breathing continues its slow, steady rhythm, consider how your own body bridges these scales. Your conscious experience, the you that thinks and feels, emerges from the collective action of trillions of cells. Each cell is itself a universe of molecular activity. Each molecule is composed of atoms, which are mostly empty space, with tiny nuclei and electron clouds. And you, in turn, are one of billions of humans on a planet, orbiting one star among hundreds of billions in a galaxy that is itself one of hundreds of billions in the observable universe. These thoughts could be overwhelming, could inspire existential anxiety, but tonight we invite them instead to inspire a kind of peaceful resignation, a recognition that you are part of something so much larger and more intricate than any worry your mind might manufacture. The same patterns that organize your cells organize the galaxies. You are made of the same substance as stars, subject to the same physical laws as everything else that exists. There is a profound democracy to this, a cosmic equality that applies equally to the struggling student, the worried parent, the anxious worker, and the peaceful sleeper. Let the concept of scale wash over you like a gentle wave. You do not need to understand it perfectly. You do not need to hold all these numbers in your head. Simply allow yourself to feel the vastness above and the intricacy below, and recognize that you, right here, right now, are the perfect size to experience both. Your consciousness is the universe, becoming aware of itself at multiple scales simultaneously. Now we are ready to begin our descent into the cellular world, carrying with us this expanded sense of perspective, this recognition that what we discover in the microscopic realm may echo with patterns from the cosmic realm. Both are real, both are happening right now, and both are, in their own way, home. Our journey begins at the boundary, as all journeys must. Before we can enter the cell, we must cross its threshold, and that threshold is the cell membrane, one of the most elegant structures in all of biology. Close your eyes for a moment and imagine yourself approaching this boundary. You have shrunk to an impossible smallness, and before you stretches a vast, shimmering wall, undulating gently like the surface of a soap bubble caught in a slow breeze. The cell membrane, also called the plasma membrane, is the structure that defines where the cell ends and the outside world begins. This might seem like a simple function, but it is anything but simple. The membrane is not a rigid wall, not a static barrier, but rather a dynamic, fluid boundary that carefully controls what enters and exits the cell. It is approximately 7 to 10 nanometers thick. A nanometer is 1/1000th of a meter, 1,000th of a micrometer. If a cell is the size of a city, the cell membrane is a city wall, only a few meters thick, yet capable of sophisticated discrimination about who and what passes through. The structure of this membrane is built on a foundation called the phospholipid bilayer. Let us take these words apart slowly, letting their meaning sink in like warmth into cold hands. A lipidus, simply a fat, a molecule that does not dissolve well in water. You have experienced lipids every time you have seen oil separate from vinegar in salad dressing. Every time you have noticed how butter resists mixing with water. Lipids are hydrophobic, a word that literally means water-fearing. Though the lipids themselves do not experience fear, they simply have a molecular structure that makes them energetically unfavorable to mix with water molecules. A phospholipid is a special type of lipid that has two parts, a head and a tail. The head contains a phosphate group, which gives this molecule its name. This head is hydrophilic, meaning water-loving, eager to interact with water molecules. The tail, on the other hand, consists of long chains of carbon and hydrogen atoms, and these chains are hydrophobic, avoiding water. So each phospholipid molecule is like a tiny being with two natures, one part attracted to water and one part repelled by it. Now imagine billions of these phospholipid molecules floating in the watery environment around a cell. Something remarkable happens, something that requires no conscious direction, no blueprint, no instruction manual. The phospholipids spontaneously arrange themselves into two layers, a bilayer with their water-loving heads pointing outward toward the watery environments on both sides of the membrane, and their water-fearing tails pointing inward toward each other, away from water. This arrangement happens automatically because it is the most energetically favorable configuration. It is what molecules do when left to follow the laws of physics and chemistry. The result is a sheet-like structure, flexible and self-healing, that forms a boundary between the cell's interior and the external world. If you could see this membrane in action, you would notice that it is not still. The phospholipid molecules drift laterally within their layer, sliding past each other like people in a crowded room, giving the membrane a fluid quality. Scientists sometimes describe this as the fluid mosaic model, a phrase that captures both the liquidity of the lipid bilayer and the fact that many other molecules are embedded in it, creating a mosaic pattern. Here we find our first echo of cosmic structure. Just as the cell membrane creates a boundary between the cell's interior and the external environment, a galaxy has boundary regions where it transitions from the dense collection of stars, gas, and dust in its main body to the sparse environment of intergalactic space. This boundary is not sharp, not a line drawn in space, but rather a gradual transition, a fading out, much like how the cell membrane is not an impermeable wall, but a selective boundary. Both structures define an inside and an outside, while allowing for exchange across the boundary. But the cell membrane is far more than just a boundary. Embedded in this phospholipid bilayer are numerous proteins, large molecules that perform specific functions. Some of these proteins span the entire width of the membrane, creating channels or tunnels through which certain molecules can pass. These are called transmembrane proteins, and they are the gates and doors in our cellular city wall. Imagine a channel protein, a tube-like structure that allows specific molecules to flow through the membrane. Some channels are always open, allowing a steady stream of particular molecules. Others are gated, meaning they open and close in response to specific signals. A sodium channel, for example, might open when it detects a certain electrical voltage across the membrane, allowing sodium ions to rush into the cell. This process is fundamental to how nerve cells transmit signals, how your brain thinks, how you perceive and respond to the world. Other proteins in the membrane act as carriers or pumps. These proteins bind to specific molecules on one side of the membrane, undergo a change in shape, and release the molecule on the other side. Some pumps require energy to operate, actively moving molecules against their concentration gradient, like pumping water uphill. The sodium-potassium pump, for instance, constantly moves sodium ions out of the cell and potassium ions into the cell, maintaining concentration differences that are essential for cellular function. This pump consumes a significant portion of the cell's energy budget, working tirelessly every moment of your life in every cell that has one. There are also receptor proteins embedded in the membrane, structures that bind to specific molecules outside the cell and then trigger changes inside the cell. These receptors are how cells sense their environment, how they receive signals from other cells, how they know when to grow, divide, or perform specialized functions. A hormone circulating in your bloodstream might bind to a receptor on a cell membrane, and that binding event might trigger a cascade of events inside the cell, ultimately changing which genes are active, which proteins are produced, how the cell behaves. The cell membrane also contains other molecules besides proteins and phospholipids. Cholesterol molecules are interspersed among the phospholipids, helping to regulate the membrane's fluidity, making it less permeable and less fluid at high temperatures, more fluid at low temperatures. Carbohydrate chains are attached to some of the proteins and lipids on the outer surface of the membrane, creating a kind of cellular identification system. These carbohydrate decorations are like molecular name tags, allowing cells to recognize each other, to distinguish self from non-self, to coordinate with neighboring cells. This selective permeability, this carefully controlled boundary, is essential for life. If the membrane were completely impermeable, nothing could enter or leave the cell, and life would be impossible. If the membrane were completely permeable, the cell's interior would immediately equilibrate with the external environment, and the cell would lose its identity, its ability to maintain the special internal conditions necessary for life. The membrane walks a perfect middle path, allowing exchange while maintaining distinction, permitting flow while preserving identity. As you lie here, feeling the weight of your body against the bed, the warmth of the blanket over you, trillions of cell membranes throughout your body are performing this delicate balancing act. Oxygen molecules are crossing membranes in your lungs, entering red blood cells. Glucose molecules are crossing membranes in your intestines, being absorbed into your bloodstream. Neurotransmitter molecules are crossing the tiny gaps between nerve cells, carrying the thoughts and sensations that constitute your conscious experience. The membrane is where the cell interfaces with reality, where inside meets outside, where the cellular universe touches the larger universe in which it exists. It is a boundary, yes, but a living boundary, an intelligent boundary, a boundary that participates actively in the life of the cell rather than simply containing it. In the galaxy, we might think of the outer regions, where stellar density decreases and the interstellar medium becomes sparser, as analogous to this boundary. Here, the galaxy transitions from its dense, central regions to the near emptiness of intergalactic space. This boundary is not passive, but active, with stars orbiting, gas flowing, and matter sometimes entering or leaving the galaxy. The comparison is not perfect because galaxies do not maintain their boundaries as actively as cells do. But the principle of a transition zone, a region where inside becomes outside, applies at both scales. Let your mind rest on this image. A membrane fluid and responsive, studded with molecular machines, separating the ordered interior of a cell from the chaos outside, allowing life to persist in a universe that tends toward disorder. Feel yourself at your own boundaries, where your skin meets the air, where your consciousness meets the world. You too are a bounded system, maintaining your identity through constant exchange with your environment, neither completely open nor completely closed, but dynamically balanced, alive. Having crossed the cell membrane, we now find ourselves floating in the cytoplasm, the gel-like substance that fills the interior of the cell. If the membrane is the boundary of the cellular world, the cytoplasm is the landscape, the medium through which everything moves, the stage on which cellular drama unfolds. Take a deep breath and imagine yourself suspended in this space, weightless, surrounded by molecules in motion. The word cytoplasm comes from Greek roots. Cyto, meaning cell, and plasm, meaning form or substance. It is the material that gives the cell its form, the substance that fills the space between the cell membrane and the nucleus, the region where most of the cell's metabolic activities occur. But the cytoplasm is not simply water, not just empty space. It is a complex, structured environment, more like a gel or a very dilute jelly than a liquid. The liquid portion of the cytoplasm is called cytosol, and it consists primarily of water with dissolved ions small molecules, and proteins. Water makes up about 70 to 80% of the cytoplasm's volume, which makes sense given that water is the medium of life on Earth, the solvent in which biological chemistry occurs. But this is not pure water, not the water you might drink from a glass. This is a solution, a mixture containing sodium ions, potassium ions, calcium ions, chloride ions, and many other dissolved substances. These ions, which are simply atoms or molecules that have gained or lost electrons and thus carry an electrical charge, are essential for numerous cellular processes. The concentrations of these ions inside the cell are carefully maintained at levels different from the concentrations outside the cell. This difference in concentration represents stored energy, like water held behind a dam. And cells use this stored energy to perform work, to send signals, to move molecules against concentration gradients. Dissolved in the cytosol are countless molecules of many types. There are amino acids, the building blocks of proteins, waiting to be assembled into larger structures. There are nucleotides, the building blocks of DNA and RNA, ready to be incorporated into genetic material or to carry energy in the form of molecules like ATP, which we will discuss later. There are sugars, lipids, vitamins, and metabolic intermediates. All the small molecules that participate in the thousands of chemical reactions that keep the cell alive. But perhaps the most impressive components of the cytosol are the proteins. Proteins are large molecules made of long chains of amino acids, folded into specific three-dimensional shapes. Each protein has a particular function determined by its shape. Some proteins are enzymes, molecules that catalyze chemical reactions, speeding them up by factors of millions or billions. Without enzymes, the chemical reactions necessary for life would occur so slowly that life as we know it would be impossible. Enzymes are the invisible workers of the cellular world, constantly cutting, joining, rearranging, and modifying other molecules. Other proteins in the cytosol serve structural roles, helping to organize the cell's interior. Still others serve as signals, as transporters, as regulators of gene expression. The cytosol is crowded with these proteins, so crowded that biochemists speak of molecular crowding as an important factor in cellular function. The concentration of macromolecules in the cytosol can be as high as 300 milligrams per milliliter, which means that a significant fraction of the cytoplasm's volume is actually occupied by dissolved molecules. This is not a dilute solution, but a concentrated one, a thick soup of biological activity. Now imagine yourself floating in this crowded molecular environment. Molecules are constantly moving, not in straight lines, but in random, jittering paths. This movement is called Brownian motion, named after the botanist Robert Brown, who first described it in the early 19th century while observing pollen grains suspended in water. Brownian motion occurs because molecules at any temperature above absolute zero are in constant thermal motion, vibrating and moving randomly. In a liquid, this means that molecules are constantly colliding with each other, changing direction, bouncing off each other in an endless, chaotic dance. At the scale of molecules in the cytoplasm, this motion is significant. A small molecule might diffuse across a typical cell in a matter of seconds, simply through random Brownian motion. This is how many molecules get where they need to go in the cell. Not through directed transport, not through conscious guidance, but through random diffusion, with concentrations naturally evening out over time, unless energy is expended to maintain concentration gradients. There is something almost meditative about this vision of molecular motion. Imagine billions upon billions of molecules, each moving randomly, colliding, bouncing, drifting, with no plan, no intention, no awareness. And yet from this chaos emerges order. Enzymes find their substrates, the molecules they act upon. Proteins find their binding partners. Signals are transmitted. Work is done. Life continues. Order emerges from randomness through the laws of probability and chemistry. The cytoplasm also contains structures that are not membrane-bound organelles, but are nonetheless distinct regions or assemblies. Ribosomes, which we will discuss in detail later, float freely in the cytoplasm or attach to other structures. The cytoskeleton, a network of protein filaments that provide structure and organization to the cell, extends throughout the cytoplasm. Various granules and inclusion bodies, temporary storage sites for nutrients or other molecules, dot the cytoplasmic landscape. When we look outward to the galaxy for comparison, the cytoplasm reminds us of the interstellar medium, the matter that exists in the space between stars. Just as the cytoplasm fills the space between the cell membrane and the nucleus, and other organelles. The interstellar medium fills the space between stars in a galaxy. This medium is not empty, but consists of gas, mostly hydrogen and helium, along with dust particles, cosmic rays, and electromagnetic radiation. The interstellar medium has different densities in different regions, just as the cytoplasm has different concentrations of molecules in different areas of the cell. In some regions of space, the interstellar medium is relatively dense, forming nebulae where new stars may eventually form. In other regions, it is extremely sparse, almost a vacuum by earthly standards, but still containing some matter. Similarly, some regions of the cytoplasm are more crowded with particular molecules or structures than others, creating functional zones within the cell. The comparison extends to movement as well. Just as molecules in the cytoplasm undergo Brownian motion, drifting randomly due to thermal energy, matter in the interstellar medium moves under the influence of various forces: gravity, radiation pressure, magnetic fields, shock waves from exploding stars. In both environments, the space between the major structures is not empty, but filled with a medium that participates in the overall function of the system, whether that system is a cell or a galaxy. There is comfort in this vision of the space between. We often focus on the structures, the organelles in a cell, or the stars in a galaxy. But the space between is equally important. It is the medium of connection, the environment in which signals propagate, the reservoir of materials, the canvas on which patterns emerge. Just as the silence between notes is essential to music, the space between structures is essential to organization. As you rest here, your own cells filled with cytoplasm, consider how much of existence is this kind of space between. Your body is mostly water, mostly the molecular equivalent of cytoplasm. The universe is mostly empty space, mostly the cosmic equivalent of interstellar medium. And yet, within these seemingly empty spaces, everything happens. Thoughts occur in the mostly water environment of your brain. Stars form in the mostly empty space of the galaxy. The space between is where life, where change, where time itself unfolds. Let yourself float in this understanding, like a molecule in cytoplasm, surrounded by activity, part of a larger whole, moving randomly, yet part of emergent order. There is no need to direct yourself, no need to control every thought. Simply let the random walk of consciousness take you where it will, trusting that, like a molecule diffusing through a cell, you will arrive where you need to be. In this case, that destination is sleep, and the path is as important as the arrival. Now we approach the most prominent landmark in the cellular landscape, the nucleus. Even from a distance, if we could see at this scale, the nucleus would dominate our view. It is the largest organelle in most cells, typically occupying about 10% of the total cell volume. A sphere, or oval structure that commands the center, or sometimes a position slightly off-center of the cellular world. The word nucleus comes from Latin, meaning kernel or core, and this is precisely what the nucleus is. The core of the cell, the command center, the repository of genetic information. If the cell is a city, the nucleus is the library and city hall combined, the place where the city's founding documents are kept, and where decisions about the city's future are made, though these decisions happen through chemistry rather than conscious thought. The nucleus is enclosed by a double membrane called the nuclear envelope. This is not a single bilayer like the cell membrane, but rather two separate phospholipid bilayers, one inside the other, with a space between them called the perinuclear space. The outer membrane of the nuclear envelope is continuous with another structure we will discuss later, the endoplasmic reticulum, creating a connected network of membranes within the cell. The nuclear envelope is not a continuous, sealed barrier. Studded throughout its surface are nuclear pores, complex structures made of many proteins arranged in an octagonal pattern around a central channel. These nuclear pores are the gateways between the nucleus and the cytoplasm, and they are remarkably sophisticated gates. Small molecules can pass through freely, diffusing in and out according to their concentration gradients. But larger molecules, particularly proteins and RNA, require specific signals to pass through the nuclear pores. Proteins that need to enter the nucleus have special sequences of amino acids called nuclear localization signals. These signals are recognized by transport proteins that guide the cargo through the nuclear pore. RNA molecules that are made in the nucleus and need to exit to the cytoplasm have their own signals and transport proteins. The nuclear pore acts as a customs checkpoint, examining molecular passports, allowing authorized travelers to pass while keeping others out. This regulation is essential because the nucleus must maintain its distinct composition, its unique environment for DNA storage and RNA synthesis. Inside the nucleus, we find the cell's genetic material, DNA, deoxyribonucleic acid, the molecule that carries the instructions for building and operating the cell. DNA is a remarkable molecule, shaped like a twisted ladder, a double helix with two strands spiraling around each other. Each strand is made of a sugar phosphate backbone with attached bases, and the bases on one strand pair with complementary bases on the other strand, forming the rungs of the ladder. There are 4 types of bases in DNA: adenine, thymine, guanine, and cytosine, often abbreviated as A, T, G, and C. Adenine always pairs with thymine, and guanine always pairs with cytosine, a complementary relationship that allows DNA to be copied accurately. The sequence of these bases along the DNA strand constitutes the genetic code, the information that determines which proteins the cell can make, and therefore what the cell can do and what it looks like. Each gene is a specific sequence of bases that codes for a specific protein or RNA molecule. The human genome, the complete set of genetic information in a human cell, contains approximately 3 billion base pairs, organized into 46 chromosomes in most cells, though sex cells have 23. But DNA in the nucleus is not simply floating freely as long, naked strands. If you could extract all the DNA from a single human cell and stretch it out in a straight line, it would be approximately 2 meters long. Yet the nucleus itself is only about 5 to 10 micrometers in diameter. How does 2 meters of DNA fit into such a tiny space? Through an extraordinary feat of molecular origami, DNA is wrapped around proteins called histones, like thread wound around spools. 8 histone proteins form a core, and DNA winds around this core approximately 1.65 times, creating a structure called a nucleosome. These nucleosomes are then further coiled and folded into higher order structures, compacting the DNA dramatically. This packaging serves multiple purposes. Obviously, it allows the DNA to fit inside the nucleus, but it also provides a mechanism for regulating gene expression, for controlling which genes are active and which are silent. DNA that is tightly packed is generally inaccessible to the proteins that read genes and make RNA copies. So tightly packed DNA is usually inactive. DNA that is more loosely packed can be accessed by these proteins and is potentially active. The cell can thus control its gene expression partly by controlling how tightly the DNA is packaged at different locations. The combination of DNA and its associated proteins is called chromatin. When the cell is not dividing, chromatin appears as a diffuse, grainy material throughout the nucleus, sometimes with denser clumps called heterochromatin, which represents tightly packed, usually inactive DNA, and lighter regions called euchromatin, which represents more loosely packed, potentially active DNA. When the cell prepares to divide, the chromatin condenses dramatically into distinct, visible structures called chromosomes, allowing the genetic material to be distributed accurately to the two daughter cells. Within the nucleus, there is often a particularly prominent structure called the nucleolus, which appears as a dense region without a surrounding membrane. The nucleolus is not an organelle in the traditional sense because it lacks a membrane, but it is a distinct functional region where ribosomal RNA is synthesized and where ribosomes begin their assembly. The nucleolus forms around clusters of genes that code for ribosomal RNA, and its size can vary depending on how actively the cell is making ribosomes. A cell that is growing rapidly and needs many ribosomes will have a large, prominent nucleolus, while a cell that is less active may have a smaller one. The nucleus is where the cell's genetic information is stored and protected. DNA is vulnerable to damage from various sources: ultraviolet radiation, reactive oxygen species produced during metabolism, errors during DNA replication. The nucleus provides a protected environment where DNA repair mechanisms can fix such damage. Multiple proteins constantly patrol the DNA, looking for abnormalities, making corrections, ensuring the integrity of the genetic information. The nucleus is also where the first step of protein synthesis occurs: transcription. When a cell needs to make a particular protein, the gene for that protein is transcribed, meaning that an RNA copy of the gene is made. This messenger RNA, or mRNA, then exits the nucleus through the nuclear pores and travels to the ribosomes in the cytoplasm, where the actual protein synthesis occurs. But the decision about which genes to transcribe, when to transcribe them, and how much mRNA to make is made in the nucleus through a complex interplay of transcription factors. Proteins that bind to DNA and regulate transcription. Here we find another compelling parallel to galactic structure. The nucleus is reminiscent of the galactic core, the central region of a galaxy where matter is most densely concentrated. At the center of most galaxies, including our own Milky Way, lies a supermassive black hole, millions or billions of times the mass of our sun. Around this black hole, stars orbit in dense concentrations, and the gravitational influence of the central region helps organize the motion of the entire galaxy. Just as the nucleus contains the information that guides cellular function, the galactic core exerts a profound influence on galactic structure and dynamics. The nucleus stores information in a stable, protected form, while the galactic core stores, in a sense, the gravitational memory of the galaxy's formation and history. Both are central command regions, though one operates through chemistry and information processing, while the other operates through gravity and motion. The nucleus, like the galactic core, is not completely isolated from the rest of the system. Materials flow in and out. Proteins enter to help package DNA, to transcribe genes, to repair damage. RNA exits to direct protein synthesis in the cytoplasm. Similarly, the galactic core exchanges matter and energy with the rest of the galaxy. Stars venture close to the core, sometimes being captured, sometimes being flung out at high speeds. Gas falls inward, and energy radiates outward. There is something deeply satisfying about this image of a protected core, a central repository of information or mass, around which the rest of the system organizes itself. It suggests that organization at any scale requires some form of center, some reference point, some concentration of information or influence. Whether that center is a nucleus containing DNA or a galactic core containing a supermassive black hole, it serves as an anchor, a source of order in an otherwise chaotic universe. As you lie here in the darkness, your own nuclei, trillions of them, are quietly maintaining the information that makes you uniquely you. Every cell nucleus in your body, with rare exceptions, contains the same genetic information, the same DNA sequence, yet different cells read different parts of this information, expressing different genes, becoming different types of cells. A nerve cell and a muscle cell have the same DNA, but read it differently, like two actors performing different roles from the same script. This is perhaps one of the most profound mysteries of biology, how a single fertilized egg with one set of genetic instructions can give rise to a complete organism with hundreds of different cell types, all working together in coordinated fashion. The answer lies partly in the nucleus, in the complex regulation of gene expression in the chemical modifications of DNA and histones that determine which genes are accessible and which are not. Let your mind rest in the nucleus for a moment, in this spherical chamber at the heart of every cell, where the instructions for life are carefully stored, where the past and future meet in the form of DNA, a molecule that records billions of years of evolutionary history while also encoding the potential for tomorrow. There is no hurry here, no urgency. The DNA replication machinery works with extraordinary fidelity, but it takes its time, carefully copying each base pair. Transcription proceeds at a steady pace, neither rushed nor delayed. In the quiet of the nucleus, information waits patiently to be read. Genes wait silently to be expressed. And the whole elaborate mechanism of heredity and cellular identity maintains itself with a kind of molecular serenity. This is the stillness at the center of activity, the calm core around which the cellular world revolves. Much as your own consciousness is the calm center around which your experience revolves, the observer at the heart of observation, awareness itself, resting now, preparing for sleep, protected and safe in its own kind of nuclear envelope. The boundaries between waking and dreaming gradually dissolving as consciousness drifts toward rest. Leaving the nucleus, we now encounter structures that float through the cytoplasm like small submarines, or, perhaps more poetically, like small planets orbiting a central sun. These are the mitochondria, the powerhouses of the cell, the organelles responsible for generating most of the cell's supply of adenosine, triphosphate, or ATP, the molecule that serves as the universal energy currency of life. The word mitochondria is actually the plural form. A single such organelle is called a mitochondrion, from Greek words meaning thread and granule, describing the appearance of these organelles when first observed under microscopes. The number of mitochondria in a cell varies tremendously depending on the cell's energy requirements. A muscle cell, which requires enormous amounts of energy to contract, might contain thousands of mitochondria. A skin cell with lower energy demands might contain only a few hundred. Some specialized cells have even more, packed so tightly that they occupy a significant fraction of the cell's volume. A mitochondrion is typically 1 to 10 micrometers long and about half a micrometer wide, roughly the size of a bacterial cell, which is not a coincidence for reasons we will explore shortly. Each mitochondrion is surrounded by two membranes, an outer membrane and an inner membrane. both phospholipid bilayers but with distinct compositions and properties. The outer membrane is relatively smooth and serves primarily as a boundary, much like the cell membrane we discussed earlier. The inner membrane is where things become interesting. This membrane is highly folded, creating structures called cristae that extend into the interior of the mitochondrion like shelves or fins. These folds dramatically increase the surface area of the inner membrane, providing more space for the molecular machines that generate ATP. The interior space enclosed by the inner membrane is called the mitochondrial matrix, a dense gel-like region filled with enzymes, ribosomes, and the mitochondrian's own DNA. Yes, mitochondria have their own DNA, separate from the DNA in the nucleus. This mitochondrial DNA is circular, like bacterial DNA, and contains genes for some of the proteins the mitochondrion needs to function. This is one of several pieces of evidence supporting the endosymbiotic theory, which proposes that mitochondria originated as free-living bacteria that were engulfed by ancient cells in a kind of permanent partnership. Over evolutionary time, most of the bacterial genes were transferred to the nucleus, but some remained in the mitochondria, making these organelles semi-autonomous entities within the cell. This origin story is worth pausing over for a moment because it illustrates one of evolution's most profound achievements: cooperation between formerly independent entities. Billions of years ago, a cell that could not generate much energy on its own incorporated a bacterium that was excellent at generating energy, and instead of digesting it, the cell allowed it to survive. The bacterium, in turn, received a protected environment and steady supply of nutrients. This partnership was so successful that it has been maintained through billions of years of evolution, and today, every cell in your body that has mitochondria is carrying on this ancient cooperation. The primary function of mitochondria is cellular respiration, the process by which organic molecules, particularly glucose, are broken down to release energy. This process occurs in several stages. First, in the cytoplasm, glucose is broken down into smaller molecules in a process called glycolysis, which generates a small amount of ATP. The products of glycolysis then enter the mitochondria, where they undergo further processing in the mitochondrial matrix. Through a series of reactions called the citric acid cycle, also known as the Krebs cycle, after the scientist who elucidated it. The citric acid cycle extracts high-energy electrons from the breakdown products of glucose and transfers them to carrier molecules called ADH and FADH2. These carrier molecules then deliver the electrons to the inner mitochondrial membrane, where the final and most productive stage of cellular respiration occurs: the electron transport chain and oxidative phosphorylation. The electron transport chain is a series of protein complexes embedded in the inner mitochondrial membrane. As electrons pass through these complexes, energy is released and used to pump protons from the mitochondrial matrix to the space between the inner and outer membranes. This creates a concentration gradient of protons, with more protons in the intermembrane space than in the matrix. The situation is similar to water held behind a dam. The concentration difference represents stored energy. At strategic locations in the inner membrane are molecular machines called ATP synthase, often described as one of the most beautiful and elegant molecular structures ever characterized. ATP synthase is like a tiny turbine, a rotating molecular motor. When protons flow back across the inner membrane, down their concentration gradient through ATP synthase, the energy of this flow is used to synthesize ATP from ADP and phosphate. The protons flow through a channel in the ATP synthase, causing part of the protein to rotate. And this rotation drives the chemical reaction that joins ADP and phosphate to create ATP. This process, called chemiosmosis, is remarkably efficient. A single glucose molecule, fully metabolized through glycolysis, the citric acid cycle, and the electron transport chain, can generate approximately 36 to 38 molecules of ATP, though the exact number varies depending on conditions. This ATP is then exported from the mitochondria to the rest of the cell, where it is used to power virtually every energy-requiring process: muscle contraction, nerve impulse transmission, protein synthesis, active transport across membranes, DNA replication, and countless others. Think of ATP as a rechargeable battery. When ATP is broken down into ADP and phosphate, energy is released, and this energy can be used to do work. The mitochondria then recharge the battery, using the energy from breaking down food molecules to convert ADP and phosphate back into ATP. This cycle of ATP breakdown and synthesis continues throughout your life, with each ATP molecule being recycled hundreds or thousands of times per day. The total amount of ATP in your body at any given moment is relatively small, perhaps only about 50 grams, yet your body synthesizes and breaks down approximately your own body weight in ATP every day. This gives you some sense of the constant intensive energy metabolism occurring in every cell. The ceaseless work of mitochondria to keep up with energy demands. When we look for cosmic parallels, mitochondria remind us of stellar nurseries, regions of galaxies where new stars are born. These stellar nurseries are dense clouds of gas and dust, where gravitational collapse and nuclear fusion generate energy, much as mitochondria are dense organelles where chemical reactions generate energy. Stars convert hydrogen into helium through nuclear fusion, releasing enormous amounts of energy in the process. Mitochondria convert glucose and oxygen into carbon dioxide and water, releasing chemical energy that is captured in ATP. Both processes involve the transformation of matter from one form to another with the release of energy. Both are essential to the larger systems they inhabit. Stars provide energy and light to their surrounding space, synthesize heavier elements that eventually become planets and life, and drive the dynamics of galaxies through their radiation pressure and eventual explosions. Mitochondria provide energy to cells, allow complex life to exist, and drive cellular processes through their ATP production. The folded inner membrane of mitochondria, the cristae, might be compared to the complex structure of stellar nurseries, with their filaments and pillars of gas and dust, their regions of varying density. Both structures maximize surface area or volume for energy generation. Both are dynamic regions where the fundamental work of existence occurs, where energy is extracted from matter and made available for use. Mitochondria also play other crucial roles in the cell beyond energy generation. They are involved in regulating calcium ion concentrations, which is important for cell signaling. They participate in certain stages of heme synthesis, the molecule that allows hemoglobin to carry oxygen in your blood. They play a role in the synthesis of certain lipids. And, somewhat paradoxically, mitochondria are also involved in triggering programmed cell death, apoptosis, when a cell is damaged beyond repair or no longer needed. In such cases, mitochondria release certain proteins that activate the cell death program, ensuring that the cell dies in a controlled manner that doesn't damage neighboring cells. This role in cell death reminds us that mitochondria are not merely passive power generators, but active participants in cellular decision-making. They sense the cell's condition, respond to stress, and can essentially vote on whether the cell should continue living. This is a profound responsibility for an organelle that began its existence as a separate organism. As you rest here, your mitochondria are working steadily, not frantically, generating ATP at a rate matched to your current needs. During sleep, your energy requirements decrease, your metabolic rate slows, and your mitochondria adjust their activity accordingly. They are remarkably responsive to cellular conditions, increasing or decreasing ATP production as needed, like power plants that ramp up or down to match electricity demand. The steady hum of mitochondrial activity is the energetic foundation of your existence. Every thought, every heartbeat, every breath depends on the ATP produced by these remarkable organelles. They work in darkness, in silence, without recognition or rest. Descendants of ancient bacteria that found a home inside ancient cells and stayed there, becoming so integrated into cellular life that they are now essential. Let yourself appreciate this constant, quiet work happening inside you right now. The chemical fires burning in millions of mitochondria throughout your body, converting the energy stored in food molecules into the energy that powers your life. There is something deeply reassuring about this. You do not have to consciously control it. Do not have to think about it or manage it. The mitochondria simply do their work as they have done for billions of years, as they will continue to do as long as you live, requiring only that you provide them with fuel and oxygen, asking nothing else in return. From the mitochondria, we now turn our attention to one of the most extensive structures in the cell, a network so vast and interconnected that it resembles a complex highway system, or perhaps the branching structure of a river delta. This is the endoplasmic reticulum, often abbreviated as ER, from Greek and Latin roots meaning within the cellular network. The endoplasmic reticulum is a continuous membrane system that extends throughout the cytoplasm, forming a series of interconnected, flattened sacs, tubes, and channels. The space inside these channels, called the lumen or cisternal space, is separated from the cytosol by the ER membrane, creating a distinct compartment within the cell where specialized chemical reactions can occur. In some cells, particularly those specialized for protein secretion, the endoplasmic reticulum can account for more than half of all the membrane in the cell, making a truly dominant structural feature. There are two types of endoplasmic reticulum, distinguished by their appearance and function: rough ER and smooth ER. These names describe exactly what you might imagine. When viewed under an electron microscope, rough ER appears studded with small particles on its cytoplasmic surface, giving it a rough or bumpy appearance. These particles are ribosomes, the molecular machines that synthesize proteins. Smooth ER lacks these attached ribosomes and appears, by comparison, smooth. Let us begin with rough ER, which is typically located near the nucleus, often continuous with the outer membrane of the nuclear envelope. In fact, the nuclear envelope can be thought of as a specialized region of the ER that is wrapped around the nucleus. The rough ER's primary function is the synthesis and modification of proteins, particularly proteins that are destined to be secreted from the cell, inserted into membranes, or transported to other organelles. When a ribosome is synthesizing a protein destined for the ER, the growing protein chain has a special sequence of amino acids at its beginning called a signal sequence. This signal sequence is recognized by a protein complex called the signal recognition particle, which pauses the translation process and directs the ribosome to the ER membrane. The ribosome then docks to a protein channel in the ER membrane, and as protein synthesis continues, the growing protein chain is threaded through this channel into the ER lumen. Inside the ER lumen, the protein undergoes several modifications. Chaperone proteins help it fold into the correct three-dimensional shape, a crucial process because a protein's function depends entirely on its shape. Enzymes in the endoplasmic reticulum may attach carbohydrate chains to the protein in a process called glycosylation, adding molecular decorations that affect the protein's stability, destination, or function. Quality control mechanisms check the protein's folding, and misfolded proteins are marked for destruction and sent back to the cytosol to be broken down. The rough ER is particularly abundant in cells that secrete large amounts of protein. For example, the cells in your pancreas that produce digestive enzymes, or the plasma cells in your immune system that produce antibodies, have extensive, rough endoplasmic reticulum networks that work continuously to generate the proteins these cells export. The rough ER is essentially a factory floor, with ribosomes as workers assembling products. The ER lumen as the processing area where products are modified and inspected, and the ER membrane as the factory building that contains and organizes the whole operation. Smooth ER, in contrast, lacks ribosomes and serves different functions. It is involved in lipid synthesis, particularly phospholipids and cholesterol, which are essential components of cellular membranes. In liver cells, smooth ER is abundant because it contains enzymes that detoxify harmful substances, including drugs and metabolic waste products. The smooth ER modifies these substances chemically, usually by adding polar groups that make them more water-soluble and easier to excrete from the body. In muscle cells, the smooth endoplasmic reticulum, often called sarcoplasmic reticulum in this context, has a specialized role in calcium storage and release. When a nerve signal tells a muscle cell to contract, the sarcoplasmic reticulum releases calcium ions into the cytosol. These calcium ions then trigger the molecular machinery of muscle contraction. When the signal ends, the sarcoplasmic reticulum pumps calcium back into its interior, allowing the muscle to relax. This rapid calcium release and uptake is essential for the rapid contraction and relaxation cycles of muscle tissue. The endoplasmic reticulum is not a static structure, but a dynamic one. Its shape and size change depending on the cell's needs. When a cell increases protein production, more rough endoplasmic reticulum forms. When steroid hormone production increases, as occurs in cells of the adrenal gland, more smooth endoplasmic reticulum develops. The ER can also move through the cytoplasm, extending new tubules, retracting others, constantly reconfiguring itself. The ER is also involved in calcium signaling throughout the cell. It stores calcium ions and releases them in response to specific signals, creating waves of calcium concentration that propagate through the cytoplasm, triggering various cellular responses. This makes the ER not just a manufacturing center, but also a signaling network, a communication system that helps coordinate cellular activities. When we consider galactic analogies, the endoplasmic reticulum reminds us of the filamentary structure of the cosmic web, the large-scale structure of the universe. On the largest scales, galaxies are not randomly distributed, but form a web-like pattern, with galaxies concentrated along filaments that stretch across hundreds of millions of light years, separated by vast voids containing few galaxies. These filaments are thought to trace the distribution of dark matter, the mysterious substance that exerts gravitational influence but does not emit light. The ER, like these cosmic filaments, forms an interconnected network that spans the cell, providing structural support and serving as a transportation system. Materials move through the ER lumen just as galaxies move along cosmic filaments. Both structures demonstrate that organization at any scale often involves networks, not just isolated objects, connections, as much as nodes, relationships, as much as entities. The rough ER, studded with ribosomes, might remind us of filaments studded with galaxies, though this is admittedly a visual comparison more than a functional one. The key point is that both systems are organized as extended networks rather than discrete, isolated structures. This network organization allows for efficient distribution of materials, rapid communication across distances, and flexible reorganization in response to changing conditions. Imagine, as you lie here in the quiet darkness, the endoplasmic reticulum in countless cells throughout your body. This vast internal network humming with activity. In your digestive system, rough ER in pancreatic cells is producing enzymes that will be secreted to help digest your last meal. In your liver, smooth ER is processing chemicals, detoxifying substances, maintaining the body's chemical balance. In your muscles, sarcoplasmic reticulum holds calcium in readiness for the next contraction, though hopefully, as you move towards sleep, your muscles are relaxing, requiring less calcium signaling, settling into stillness. The ER is like the cell's internal internet, a network that connects different regions, allows information and materials to flow, provides specialized services in different areas, maintains the infrastructure of cellular life. It is easy to overlook in discussions of cell biology because it is not as conceptually simple as a nucleus or mitochondria, not as easy to assign a single clear function. But the ER's very complexity, its multifunctionality, its network structure, makes it essential to complex cellular life. Without the ER, cells could not efficiently produce and process the thousands of different proteins they need, could not synthesize membranes, could not detoxify harmful substances, could not regulate calcium signaling. The ER is where much of the cell's chemical transformation occurs, where raw materials become finished products, where information encoded in genes becomes functional molecules that actually do the work of life. Rest in this image of networks within networks, cells connected by membranes just as galaxies are connected by filaments of dark matter, just as your thoughts are connected by networks of neurons, just as all things are connected by the fundamental forces of nature. Separation is an illusion, or at least a simplification. Everything is connected. Everything is networked. Everything flows into everything else. And the boundaries we draw are convenient fictions that help us think about systems too complex to grasp in their entirety. Let this connectedness ease your mind, dissolve your sense of isolation, remind you that you are part of vast networks at every scale, from the molecular to the cosmic, and that these networks support you, sustain you, connect you to everything that exists. Our journey through the cellular landscape now brings us to a structure that resembles a stack of pita bread, or perhaps a neat pile of pancakes. This is the Golgi apparatus, named after Camillo Golgi, the Italian physician and scientist who first described it in the late 19th century. For many years, some scientists doubted that the Golgi apparatus was even real, thinking it might be an artifact of the staining techniques used to visualize cells. But eventually, with better microscopy, its reality was confirmed, and today we understand it as an essential organelle involved in processing and shipping cellular products. The Golgi apparatus, sometimes called the Golgi complex or Golgi body, consists of a stack of flattened membrane sacs called cisternae. A typical Golgi stack contains 4 to 6 cisternae, though the number can vary. Each cisterna is like a flattened balloon with a narrow lumen inside, and the cisternae are stacked close together but remain separate, not fused into a single compartment. The Golgi stack has directionality, meaning that the two faces of the stack are different and serve different functions. The cis face, or receiving side, is oriented toward the endoplasmic reticulum. Here, vesicles carrying newly synthesized proteins and lipids from the ER arrive, fuse with the Golgi membrane, and deliver their cargo. The transface, or shipping side, is oriented toward the cell membrane or other parts of the cell. From here, vesicles bud off carrying finished products to their final destinations. Between these two faces, in the medial cisternae, the actual processing occurs. As proteins move through the Golgi stack from cis to trans, they undergo a series of modifications. Enzymes in different cisternae modify the carbohydrate chains that were added to proteins in the ER, trimming some sugars, adding others, creating specific carbohydrate structures that serve as molecular addresses or identification tags. These modifications are not random but highly specific. Different enzymes reside in different cisternae, and the sequential movement of proteins through the stack ensures that modifications occur in the correct order. It is like an assembly line, but instead of car parts being added, molecular decorations are being modified. And instead of cars moving along a conveyor belt, proteins are being transported through a stack of membrane compartments. The Golgi also sorts proteins based on their destinations. Some proteins are destined for the cell membrane, where they will function as receptors, channels, or structural components. Others are destined for secretion from the cell. Still others are destined for lysosomes, the cell's digestive organelles that we will discuss shortly. Each destination requires different molecular signals, and the Golgi reads these signals and packages proteins into appropriate vesicles. This sorting function is remarkably precise. The Golgi must distinguish among dozens or hundreds of different proteins, determine the correct destination for each, and ensure they are packaged into the right vesicles with the right membrane composition and the right additional proteins that will guide the vesicle to its destination. It is like a postal sorting facility, but operating at the molecular level, handling millions of packages per hour with an error rate that must be extraordinarily low, because mistakes in protein trafficking can lead to cellular dysfunction or disease. The process by which materials move through the Golgi stack is still debated by cell biologists. One model, the vesicular transport model, proposes that the cisternae themselves are stable and that small vesicles carry cargo from one cisterna to the next. Another model, the cistern almaturation model, proposes that the cisternae themselves move and mature, with the ciscisterna gradually becoming medial and then trans as it moves through the stack, while new ciscisterna form from ER-derived vesicles. Evidence exists for both mechanisms, and it is possible that both occur, perhaps in different cell types or under different conditions. Regardless of the precise mechanism, the result is clear. Proteins entering the cis face of the Golgi emerge from the trans face, modified, sorted, and packaged for their final destinations. Vesicles bud from the trans face, constantly each one carrying a specific cargo to a specific destination, each one wrapped in a piece of membrane that will fuse with the membrane of the target compartment or with the cell membrane itself. When proteins destined for secretion reach the cell membrane, their vesicles fuse with the membrane in a process called exocytosis, releasing the vesicle contents outside the cell. This is how your pancreatic cells release digestive enzymes into your digestive tract, how your nerve cells release neurotransmitters to communicate with other nerve cells, how many hormones and signaling molecules are released to affect distant tissues. The Golgi apparatus is also involved in the synthesis of certain complex carbohydrates and in the modification of lipids. In plant cells, the Golgi plays a crucial role in cell wall synthesis, producing the complex polysaccharides that form the rigid walls surrounding plant cells. in cells specialized for secretion, such as goblet cells in the intestines that secrete mucus. The Golgi is extraordinarily well developed, occupying a large fraction of the cell's volume. For our purposes, comparing cellular structures to galactic ones, the Golgi apparatus reminds us of the processing that occurs in molecular clouds before stars form, or perhaps the way matter is processed as it falls into the accretion disk around a black hole. Just as proteins are modified as they pass through the Golgi, matter is transformed as it undergoes various physical processes in space. Gas in molecular clouds is compressed, heated, cooled, mixed with dust, enriched with heavier elements from previous generations of stars, all before it eventually collapses to form new stars. The Golgi's function as a sorting and shipping center might also remind us of how matter and energy are distributed in a galaxy, not uniformly, but in organized ways. Stars form in certain regions, supernova remnants spread enriched material, radiation from the galactic core influences the surrounding medium. There is a kind of directed flow, a processing and distribution that characterizes both the Golgi in a cell and material flow in a galaxy. But perhaps the most interesting parallel is the idea of modification over time. As proteins move through the Golgi, they are progressively modified, their final form determined by the sequential action of multiple enzymes. Similarly, as matter moves through different regions of a galaxy, it is transformed by different physical processes: radiation, shock waves, gravitational compression, magnetic fields. In both cases, the history of where something has been determines what it becomes. This concept applies even to your own experience as you lie here approaching sleep. Your consciousness at this moment is the result of a lifetime of experiences, a sequential processing of sensory input, learning, memory formation, emotional reactions. You have been modified by everything you have encountered, and you continue to be modified with each passing moment. Your thoughts right now are like proteins moving through the Golgi, being processed, sorted, some being sent to memory storage, others being discarded, still others being prepared for expression in words or actions tomorrow. The Golgi apparatus works constantly but quietly without fanfare, performing essential tasks that keep the cell functioning. If the Golgi fails, proteins are not properly sorted. Cellular compartments receive the wrong materials. The cell's organization breaks down. Several diseases are caused by defects in Golgi function or in the trafficking pathways that move materials to and from the Golgi. The smooth operation of this organelle is essential to cellular health, yet it receives little attention in popular discussions of biology, overshadowed by the more dramatic nucleus or the more obviously essential mitochondria. But here, in this quiet moment before sleep, we can appreciate the Golgi for what it is, a marvel of molecular organization, a sorting and processing center that handles complexity with elegance and efficiency, a reminder that systems require not just parts, but also logistics, not just production, but also distribution. The cell, like any complex organization, needs infrastructure for handling materials, for ensuring that products reach their destinations, for maintaining order in the face of entropy. Let the thought of the Golgi apparatus, stacks of membranes processing and sorting molecular cargo, help your own mind sort through the day's experiences, filing memories, discarding irrelevancies, preparing for tomorrow while allowing today to fade into the past. Trust in the processing, in the sorting, in the gradual movement from intake to output, from experience to memory, from waking to sleeping. We now descend into what might be called the cell's recycling and waste management system, a set of organelles that perform the somewhat unglamorous but absolutely essential function of breaking down unwanted materials. These are the lysosomes and peroxisomes, both small membrane-bound compartments that contain powerful enzymes capable of degrading various molecules. Lysosomes are often described as the cell's stomach or digestive system, which captures their function, if not their elegance. Each lysosome is surrounded by a single membrane and filled with an acidic interior environment, typically with a pH around 4.5 to 5.0, compared to the cytoplasm's pH of around 7.2. This acidity is maintained by protein pumps in the lysosomal membrane that actively transport protons into the lysosome's interior, using energy in the form of ATP. Why maintain such an acidic environment? Because the enzymes inside lysosomes, called acid hydrolases, function optimally at acidic pH. There are approximately 50 different types of acid hydrolases in a typical lysosome, each capable of breaking down different types of molecules. Proteases break down proteins into amino acids. Lipases break down lipids. Nucleases break down nucleic acids, and so on. Together, these enzymes can degrade essentially any biological molecule the cell might need to dispose of. This is potentially dangerous. If these destructive enzymes were released into the cytoplasm, they could begin digesting the cell's own proteins, lipids, and nucleic acids, effectively killing the cell. The lysosomal membrane protects the cytoplasm from these enzymes, keeping them safely contained. Additionally, even if the lysosomal membrane were to rupture, the acid hydrolases would be largely inactive in the cytoplasm's neutral pH, providing a backup safety mechanism. Lysosomes digest materials that come from two main sources: materials brought into the cell from outside through endocytosis, and the cell's own worn-out organelles and macromolecules. The process of breaking down the cell's own components is called autophagy, from Greek words meaning self-eating. This might sound destructive, but autophagy is actually a crucial maintenance process that allows cells to renew themselves, breaking down old, damaged, or unnecessary components and recycling their molecular building blocks. Imagine a mitochondrion that has been working for weeks or months and is now damaged, its membranes compromised, its DNA accumulating mutations. The cell recognizes this damaged mitochondrion and engulfs it in a double membrane structure called an autophagosome. This autophagosome then fuses with a lysosome, forming an autolysosome, and the lysosomal enzymes break down the mitochondrion completely, releasing amino acids, nucleotides, lipids, and other molecules that can be reused to build new cellular components. This recycling is continuous. Cells are constantly breaking down and rebuilding themselves, replacing old components with new ones, responding to damage, adapting to changing conditions. In fact, most of the molecules in your body are replaced regularly. The atoms that compose your body today are not the same atoms that composed it a year ago. You are like a river, maintaining your form even as your substance constantly flows and changes. Lysosomes also digest materials brought into the cell through endocytosis. When a cell engulfs a particle or a droplet of extracellular fluid, it forms an endocytic vesicle that can fuse with a lysosome. The lysosomal enzymes then break down the contents of the vesicle. This is particularly important for cells of the immune system, such as macrophages, which engulf bacteria and other pathogens. The pathogen is brought into the cell in an endocytic vesicle, which fuses with a lysosome, and the pathogen is destroyed by the lysosomal enzymes. Peroxisomes are similar to lysosomes in size and in being membrane-bound compartments containing degradative enzymes, but they serve different functions. Peroxisomes contain enzymes that break down fatty acids through a process called beta oxidation, and enzymes that break down hydrogen peroxide, a toxic byproduct of various metabolic reactions. The name peroxisome comes from this latter function. These organelles contain peroxide and contain the enzyme catalase that breaks peroxide down into water and oxygen. Hydrogen peroxide is continuously produced in cells as a byproduct of various oxidation reactions. And it is dangerous because it can damage DNA, proteins, and lipids through oxidation. Peroxisomes prevent this damage by containing and breaking down hydrogen peroxide quickly. The enzyme catalase is extraordinarily efficient, one of the fastest enzymes known, capable of breaking down millions of hydrogen peroxide molecules per second. Peroxisomes are also involved in the synthesis of certain lipids, particularly plasmalogens, which are important components of myelin, the insulating sheath around nerve cells. In the liver, peroxisomes help detoxify various harmful substances, much like smooth ER does, providing a backup or complementary detoxification system. Both lysosomes and peroxisomes exemplify the cell's need for compartmentalization. By isolating dangerous chemical reactions in membrane-bound organelles, the cell can harness powerful chemical processes without risking damage to itself. This is a general principle in cell biology. Dangerous or incompatible processes are separated into different compartments, each with its own specialized environment. When we look for parallels in galactic structure, lysosomes and peroxisomes remind us of the processes by which galaxies recycle material. When massive stars explode as supernovae, they break apart the heavy elements that were synthesized in their cores, scattering them through the interstellar medium. This recycled material, enriched with elements heavier than hydrogen and helium, can then be incorporated into new generations of stars and planets. In this sense, supernovae serve a function analogous to autophagy, breaking down old structures and making their components available for new construction. The interstellar medium itself acts as a kind of recycling system, mixing material from different sources, allowing gas to cool and condense to form new stars, or heating and dispersing gas through shocks and radiation, just as lysosomes break down complex molecules into simple building blocks. Astrophysical processes break down complex structures into simpler components. that can be reorganized. There is something almost philosophical about this recognition that destruction is essential to creation, that breaking down is as important as building up, that recycling is not an unfortunate necessity but a fundamental aspect of how complex systems maintain themselves over time. Nothing lasts forever. Everything wears out, becomes damaged, becomes obsolete. The elegant solution is not to mourn this fact, but to build systems that turn endings into beginnings, that convert waste into resource, that make impermanence sustainable. Your own body demonstrates this constantly. The cells lining your digestive tract are replaced every few days. Your red blood cells last about 4 months before being broken down and replaced. Even your bones, seemingly permanent, are constantly being remodeled, broken down by cells called osteoclasts, and rebuilt by cells called osteoblasts. You are in a state of constant renewal, constant recycling, constant becoming. As you lie here feeling sleep approach, your cells are conducting their quiet work of breakdown and renewal. Lysosomes are digesting worn-out organelles. Peroxisomes are neutralizing hydrogen peroxide. And everywhere in your body, the old is making way for the new. This is not something to resist or regret, but something to accept, even appreciate. You are not a fixed, static entity, but a dynamic process, a pattern that maintains itself through change, a dance that continues even as the dancers change. Let this understanding ease any anxiety about impermanence, about aging, about change. You are designed for change. Your cells know how to let go of what is no longer useful, how to break down what is broken, how to recycle components for new purposes. Trust in this process, in this cellular wisdom that knows without knowing, that maintains without maintaining, that renews without effort or intention, simply by following the laws of chemistry and the logic of life. Rest now in the knowledge that even in breakdown there is continuation, even in destruction there is creation, even in endings there are beginnings. The lysosomes work in darkness, in acid, in isolation, performing unpleasant tasks with perfect efficiency, asking nothing in return, making renewal possible through their willingness to destroy. There is beauty in this if we have eyes to see it, if we have the wisdom to appreciate function over appearance, process over permanence. Having explored the membrane-bound organelles, we now turn our attention to a different type of cellular structure, one that is not enclosed in membranes, but instead forms an intricate three-dimensional network throughout the cell. This is the cytoskeleton, the structural framework that gives cells their shape, allows them to move, provides tracks for intracellular transport, and helps organize the cell's interior. The word cytoskeleton suggests something rigid and static, like the skeleton in your body, and indeed the cytoskeleton does provide structural support. But unlike bones, which change slowly if at all, the cytoskeleton is dynamic, constantly assembling and disassembling, reorganizing itself in response to cellular needs. It is more like a tent frame that can be quickly erected or collapsed than like the steel frame of a building. The cytoskeleton consists of three main types of protein filaments, each with different properties and functions. The thinnest filaments are called microfilaments or actin filaments, approximately 7 nanometers in diameter. These are made of a protein called actin, one of the most abundant proteins in many cell types. Actin filaments are particularly important for cell movement and for changes in cell shape. Actin molecules can assemble into long filaments, with molecules adding to one end and dissociating from the other, creating what is called a treadmilling effect, where the filament essentially moves, even though it maintains roughly the same length. Actin filaments are often found concentrated at the cell's edge, where they help push the membrane forward during cell movement. When a cell like a white blood cell crawls toward a site of infection, actin polymerization at the leading edge pushes the membrane forward, creating protrusions called lamellipodia or filopodia. Actin filaments also work with motor proteins, particularly myosin, to generate force. In muscle cells, the interaction between actin filaments and myosin filaments is what causes contraction. Myosin molecules walk along actin filaments, using energy from ATP to change shape in a way that produces force and movement. When many myosin molecules do this simultaneously, they can slide actin filaments past each other, causing the overall structure to contract. This is the basis of all muscle contraction. From the beating of your heart to the movement of your eyes as you read, from the peristalsis of your digestive tract to the voluntary movements of your arms and legs. The intermediate filaments are medium-sized, approximately 10 nanometers in diameter, and serve primarily structural functions. Unlike actin filaments and microtubules, which we will discuss next, intermediate filaments are more stable less dynamic. They provide mechanical strength to cells, helping them resist stress and strain. Different types of cells have different types of intermediate filaments. Keratin filaments are abundant in skin cells, providing the toughness that protects your body's surface. Neurofilaments help maintain the long, thin shape of nerve cell axons. Nuclear lamins form a network just inside the nuclear envelope, providing structural support to the nucleus. The thickest components of the cytoskeleton are microtubules, approximately 25 nanometers in diameter. Hollow tubes made of a protein called tubulin. Microtubules are highly dynamic, growing rapidly at one end while shrinking at the other, exhibiting a behavior called dynamic instability. A microtubule can switch rapidly between growing and shrinking phases, allowing quick reorganization of the cytoskeletal network. Microtubules serve as tracks for intracellular transport. Motor proteins called kinesins and dyneins walk along microtubules, carrying cargo such as vesicles, organelles, or protein complexes from one part of the cell to another. Kinesins generally move toward one end of the microtubule, typically the end near the cell membrane, while dyneins move toward the other end, typically the end near the cell center. This bidirectional transport system allows efficient movement of materials throughout the cell, much faster than diffusion alone would permit. During cell division, microtubules form the mitotic spindle, the structure that separates chromosomes into the two daughter cells. The spindle is a beautiful and complex arrangement of microtubules, radiating from two poles, attaching to chromosomes at their centromeres, and pulling them apart as the cell divides. The precision of this process, ensuring that each daughter cell receives exactly the right set of chromosomes, depends critically on the proper function of microtubules and their associated proteins. Microtubules also form the core of cilia and flagella, hair-like structures that extend from some cell surfaces. Cilia are short and numerous, beating in coordinated waves to move fluid past the cell surface. The cells lining your respiratory tract have cilia that beat rhythmically, sweeping mucus and trapped particles up and out of your lungs, a cleaning mechanism that protects you from inhaled contaminants. Flagella are longer and typically occur singly or in small numbers, propelling the cell through fluid. The most familiar example is the sperm cell, which uses its flagellum to swim toward the egg during fertilization. The cytoskeleton is not just a passive scaffold, but an active, responsive system. Cells can reorganize their cytoskeleton in response to external signals, changing shape, moving, dividing, forming specialized structures. The rapid assembly and disassembly of cytoskeletal filaments allows this flexibility. Within minutes, a cell can completely remodel its cytoskeleton, changing from a rounded shape to a spread out shape, or extending protrusions in a new direction. For our purposes of finding cosmic parallels, the cytoskeleton reminds us of the dark matter scaffolding thought to underlie the large-scale structure of the universe. Dark matter, though invisible and still mysterious, is believed to form a cosmic web of filaments, along which visible matter, including galaxies, is organized. The distribution of galaxies across the universe is not random, but follows this underlying structure, with galaxies forming along filaments at the intersections of filaments in a pattern that reflects the distribution of dark matter. Similarly, the cytoskeleton provides an underlying structure that organizes the cell's interior, determines its shape, and guides the movement of organelles and vesicles. Just as galaxies are distributed along dark matter filaments, organelles are positioned and transported along cytoskeletal filaments. Both systems demonstrate that organization requires structure, that complexity needs scaffolding, that patterns emerge from underlying frameworks. The dynamic nature of both systems is also worth noting. The cosmic web is not static. Galaxies move along filaments. Matter flows. Structures evolve over cosmic time. Similarly, the cytoskeleton is constantly reorganizing. Filaments growing and shrinking, networks forming and dissolving. Both systems balance stability and change, maintaining structure while allowing reorganization, providing framework while permitting flow. As you rest here, your cytoskeleton throughout your body is working constantly. In your muscle cells, actin and myosin filaments slide past each other, even in the tiny, unconscious contractions that maintain muscle tone. In your nerve cells, motor proteins are walking along microtubules, transporting neurotransmitter vesicles from the cell body where they are made to the axon terminal, where they will be released. In cells throughout your body, intermediate filaments provide the structural integrity that prevents cells from falling apart under mechanical stress. The cytoskeleton is invisible to ordinary perception, yet essential to every movement. Every change in cell shape, every instance of intracellular transport. It is the hidden architecture of life, the framework that makes everything else possible. Without it, cells would collapse into formless blobs, unable to maintain shape, unable to divide, unable to move, unable to organize their interiors. Let yourself appreciate this hidden structure, this framework that supports without being seen, that organizes without being obvious. Your own life, too, has its invisible structures, its habits and patterns and relationships that provide framework and support, that allow you to maintain form even as your experiences and thoughts constantly flow and change. Some of these structures are rigid, like intermediate filaments, providing stable support. Others are dynamic, like actin filaments and microtubules, allowing quick reorganization when needed. As sleep approaches, feel your own structure relaxing, your cytoskeleton in some sense releasing its tension, your muscles letting go, your body settling into the bed's support. You do not need to hold yourself together consciously. The structures maintain themselves. The frameworks persist. The organization continues even as consciousness fades. Trust in structure. Trust in framework. Trust in the invisible scaffolding that supports existence at every scale, from cytoskeleton to cosmic web, from the architectural framework of cells to the architectural framework of galaxies. Until now, we have been exploring cellular structures common to most eukaryotic cells, the cells that make up animals, plants, fungi, and many single-celled organisms. But now we must acknowledge significant differences, particularly the specializations found in plant cells. These differences are not trivial, but reflect fundamentally different lifestyles, different evolutionary solutions to the challenges of existence. The most obvious difference between plant and animal cells is the presence of a cell wall in plant cells. This wall surrounds the cell membrane, external to it, providing rigid structural support. The cell wall is made primarily of cellulose, a polysaccharide, a long chain of glucose molecules linked together in a specific way that makes the resulting fiber extraordinarily strong. Cellulose fibers are embedded in a matrix of other polysaccharides and proteins, creating a composite material that is both strong and somewhat flexible, like fiberglass or reinforced concrete. The cell wall serves multiple functions. Most importantly, it allows plant cells to develop turgor pressure, the pressure exerted by the cytoplasm pushing against the cell wall from inside. This pressure occurs because plant cells actively accumulate water, which enters by osmosis, attracted by the high concentration of dissolved substances in the cell's interior. As water enters, the cell swells, but the cell wall prevents unlimited swelling. Instead, pressure builds up inside, and this pressure provides structural support to the plant. This is why plants wilt when deprived of water. Without adequate water, turgor pressure drops, and cells can no longer support themselves. The cell walls are still there, still strong, but without internal pressure pushing against them. They cannot maintain the plant's structure. When water is restored, cells take up water again. Turgor pressure returns, and the plant stands upright once more. This is a very different structural strategy from what animals use. Animals have rigid skeletons, or, in the case of invertebrates like worms, maintain shape through muscular contraction and hydrostatic pressure in fluid-filled cavities. The cell wall, because it is relatively rigid and strong, allows plants to grow to great heights. The tallest trees, over 100 meters tall, are able to reach such heights, partly because their cells have walls that can support the weight of all that mass. But the cell wall also limits plant cells in certain ways. Plant cells cannot change shape as easily as animal cells, cannot move as freely, cannot engulf particles through phagocytosis. Plants have evolved different solutions to these challenges, solutions that work within the constraints imposed by having cell walls. Another major difference is the presence of chloroplasts in plant cells' organelles that conduct photosynthesis. Chloroplasts are similar in some ways to mitochondria. They are enclosed by double membranes. They have their own DNA, and they are thought to have originated as free-living bacteria that entered into endosymbiotic relationships with ancient eukaryotic cells, just as mitochondria did, though in a separate event. A chloroplast is typically 5 to 10 micrometers long, roughly the size of a bacterial cell, and shaped like a lens or a flattened sphere. Inside the chloroplast, suspended in a gel-like matrix called the stroma, are stacks of flattened membrane sacs called thylakoids. These thylakoid stacks, called grana, are where the light-dependent reactions of photosynthesis occur. The thylakoid membranes contain chlorophyll, the green pigment that absorbs light energy, and the protein complexes that convert light energy into chemical energy. Photosynthesis is essentially the reverse of cellular respiration. while cellular respiration breaks down glucose and releases energy. Photosynthesis uses energy from sunlight to build glucose from carbon dioxide and water. The overall equation is deceptively simple. 6 carbon dioxide molecules plus 6 water molecules plus light energy yields 1 glucose molecule plus 6 oxygen molecules. But the actual process is complex, involving dozens of enzymes and protein complexes, multiple electron transport chains, and intricate regulation. The light-dependent reactions occur in the thylakoid membranes. Light strikes chlorophyll molecules, exciting electrons to higher energy levels. These energized electrons are passed through an electron transport chain, similar to the one in mitochondria, but working in the opposite direction. As electrons move through this chain, protons are pumped across the thylakoid membrane, creating a concentration gradient. These protons then flow back through ATP synthase, the same molecular motor found in mitochondria, generating ATP. The electrons eventually reduce a molecule called NADP+ to NADPH, creating another energy carrier. The light-independent reactions, also called the Calvin cycle, occur in the stroma. Here, the ATP and NADPH produced by the light reactions are used to convert carbon dioxide into glucose through a series of enzymatic reactions. The key enzyme, rubisco, is probably the most abundant protein on Earth, present in huge quantities in every photosynthetic organism. Rubisco captures carbon dioxide from the air and attaches it to a 5-carbon sugar, initiating the process that ultimately produces glucose. Chloroplasts are literally solar panels, devices for capturing light energy and converting it into chemical form. Nearly all the energy that flows through ecosystems on Earth ultimately comes from photosynthesis. Plants capture sunlight and store it in organic molecules. Animals eat plants, accessing that stored energy. Carnivores eat herbivores, transferring energy up the food chain. Even fossil fuels, coal and oil, represent ancient sunlight captured by photosynthetic organisms millions of years ago and compressed and transformed by geological processes. When we think about cosmic parallels, chloroplasts remind us that stars too are the energy sources for their surroundings. The sun floods Earth with light, and chloroplasts capture a tiny fraction of that light, enough to power the entire biosphere. Similarly, stars flood their galactic neighborhoods with light and other radiation, providing energy that drives various astrophysical processes, heating gas clouds, ionizing atoms, exerting radiation pressure. Plant cells also typically have a large central vacuole, a membrane-bound compartment that can occupy up to 90% of the cell's volume in mature plant cells. This vacuole is filled with cell sap, a watery solution containing various dissolved substances, sugars, salts, pigments, waste products. The vacuole serves multiple functions. It provides much of the turgor pressure that supports the cell, since the vacuole membrane, called the tonoplast, allows water to enter, but retains dissolved solutes, creating an osmotic situation that draws water in. The vacuole also serves as a storage compartment for nutrients, particularly sugars and amino acids, that the plant may need later. It can store pigments like the red, purple, and blue anthocyanins that color many flowers and fruits. It can sequester waste products or toxic substances, keeping them away from the cytoplasm where they might interfere with cellular processes. And it can help maintain cytoplasmic pH and ionic composition by taking up or releasing ions as needed. The large central vacuole is part of why plant cells can grow to large sizes without requiring proportionally more cytoplasm. As a plant cell matures, its vacuole expands, filling with water, and the cell grows. But most of this growth is just water in the vacuole, requiring relatively little energy or resources to produce. The cytoplasm remains as a relatively thin layer between the vacuole and the cell membrane, but this is sufficient for the cell's metabolic needs. Plant cells are also connected to neighboring cells through plasmodesmata, narrow channels that pass through the cell walls and connect the cytoplasm of adjacent cells. These channels allow direct transfer of small molecules and even some proteins between cells, creating a kind of cytoplasmic continuity across the plant. In a sense, all the cells in a plant are interconnected, forming a supercellular organism where materials can flow from cell to cell without having to cross cell membranes. These specializations, the cell wall, chloroplasts, large central vacuole, and plasmodesmata, make plant cells fundamentally different from animal cells, adapted for a fundamentally different lifestyle. Plants are autotrophic, meaning they produce their own food through photosynthesis. They are sessile, meaning they cannot move from place to place. They have evolved to thrive in these conditions, to compete not through movement or predation, but through efficient capture of sunlight, through growth toward light, through the production of vast numbers of seeds that can disperse to new locations. As you rest here in darkness, plants across the world are either conducting photosynthesis if they are in sunlight, or waiting for dawn, maintaining themselves through cellular respiration of stored sugars, just as you are maintaining yourself through cellular respiration of your last meal. The plants outside your window, if there are any, are performing their silent work of converting carbon dioxide into oxygen, of building organic matter from air and water and sunlight, of participating in the vast cycles of matter and energy that make Earth a living planet. There is something calming about the existence of plants, about their steadiness, their patience, their simple strategy of staying in one place and growing toward the light. They do not worry, do not plan, do not regret. They simply grow when conditions permit, rest when conditions require, produce flowers and fruits at the appropriate time, all according to patterns encoded in their genes and triggered by environmental cues. They are perfectly adapted to their way of life, asking nothing more than light, water, carbon dioxide, and minerals from the soil. Let the thought of plants, quiet and patient, help calm your mind. You need not be constantly active, constantly moving, constantly doing. Like a plant in darkness, you can simply rest, maintaining yourself through basic metabolism, waiting for the dawn of tomorrow, trusting that when light returns, you will be ready to grow again, to photosynthesize metaphorically, to capture energy and build structure. For now, rest is enough. Maintenance is enough. Simply being is enough. Returning now to animal cells, we can appreciate their differences from plant cells, the adaptations that suit them for their particular lifestyle. Animal cells, lacking cell walls, are typically smaller and more flexible than plant cells. They can change shape more easily, allowing for phenomena like cell movement, phagocytosis, and the dramatic shape changes that occur during cell division. One structure present in most animal cells but absent in most plant cells is the centriole. Centrioles are cylindrical structures approximately 0.4 micrometers long, composed of 9 triplets of microtubules arranged in a circle, creating a pinwheel or cartwheel appearance when viewed from one end. Animal cells typically have 2 centrioles, oriented perpendicular to each other, forming a structure called the centrosome. The centrosome serves as the microtubule organizing center of the cell. Microtubules grow from the centrosome, radiating outward like spokes from a wheel hub. During cell division, the centrosome duplicates, and the two centrosomes move to opposite ends of the cell, where they help organize the mitotic spindle. The mitotic spindle's microtubules attach to chromosomes and pull them apart, ensuring that each daughter cell receives the correct set of chromosomes. Plant cells lack centrioles, but still form mitotic spindles during cell division, suggesting that centrioles are not absolutely required for spindle formation. Other microtubule organizing structures serve similar functions in plant cells, but in animal cells, centrioles are the primary organizers of microtubule arrays, and their position often determines the overall organization of the cell's cytoskeleton. Animal cells also differ from plant cells in their approach to providing structural support. Without cell walls and turgor pressure, animal cells use their cytoskeleton more extensively for structural support. The intermediate filaments we discussed earlier are particularly important, forming networks that give cells mechanical strength. Additionally, animal cells often have more elaborate arrangements of actin filaments near the cell membrane, creating a cortical network that helps maintain cell shape. Many animal cells are specialized in ways that plant cells are not, reflecting the greater diversity of tissue types in animals. Nerve cells, or neurons, are perhaps the most dramatically specialized cells, with long extensions called axons that can be over a meter long in large animals, allowing electrical signals to be transmitted rapidly over long distances. Muscle cells are filled with actin and myosin filaments arranged in regular patterns that allow powerful contraction. Red blood cells lose their nuclei during development, becoming essentially bags of hemoglobin optimized for oxygen transport, with no ability to reproduce or repair themselves but able to squeeze through the narrowest capillaries. Animal cells, being heterotrophic, meaning they cannot produce their own food through photosynthesis, must obtain organic molecules by consuming other organisms. This necessity has driven the evolution of digestive systems, specialized cells and tissues that break down food, absorption systems that take up nutrients, and circulatory systems that distribute nutrients to cells throughout the body. The diversity of animal cell types reflects this complexity, with different cells specialized for digestion, absorption, circulation, sensation, movement, reproduction, immune defense, and many other functions. Animal cells typically have many small vacuoles rather than one large central vacuole. these vacuoles serve various functions. Some are involved in endocytosis, bringing material into the cell. Others are specialized for storing specific substances, but none approaches the size of a plant cell's central vacuole. And animal cells rely less on vacuoles for structural support. The flexibility of animal cells, their ability to change shape and move, has enabled the evolution of animal motility, the ability to move from place to place. This, in turn, has enabled entirely different ecological strategies compared to plants. Animals can pursue prey, flee from predators, migrate to favorable environments, find mates, and actively disperse their offspring. This mobility has driven the evolution of nervous systems to coordinate movement, sensory systems to navigate and perceive the environment, and complex behaviors. When we compare animal cells to plant cells in the context of galactic structures, we might note that both types of cells, despite their differences, follow similar organizational principles. Both have membranes defining compartments. Both have cytoplasm as an internal medium. Both have nuclei storing genetic information. Both have mitochondria generating ATP. Both have elaborate internal structures. The differences are variations on common themes, adaptations to different lifestyles, much as different types of galaxies, spiral, elliptical, irregular, are variations on the theme of gravitationally bound collections of stars, gas, and dust. The existence of both plant and animal cells, both sessile and motile, organisms, both autotrophs and heterotrophs, enriches the biosphere, creates more ecological niches, enables more complex food webs. Similarly, the diversity of galactic types enriches the cosmos, creates variation in star formation rates and chemical evolution, enables a more complex universe. As you lie here, your animal cells with their specialized features are maintaining your body, your nervous system, processing sensory information, even in the dimness of near sleep. Your muscle cells holding you in position or relaxing to allow comfortable rest. Your red blood cells circulating through capillaries delivering oxygen. Your white blood cells patrolling for infections. This cooperation of specialized cells creates you a multicellular organism vastly more complex than any individual cell, yet ultimately built from cells, dependent on cellular function. For every aspect of your existence, The difference between plant and animal cells reminds us that there is no single right way to be cell, no single correct design. Evolution has explored many solutions, many adaptations, many strategies. Some lineages developed cell walls and photosynthesis, becoming plants. Others retained flexibility and developed motility, becoming animals. Still others, the fungi, developed cell walls but remained heterotrophic, absorbing nutrients from their environment. Each approach works. Each has been successful. Each has filled Earth with descendants. This diversity of solutions, this exploration of possibility space, characterizes not just cellular evolution, but evolution at all levels, perhaps even cosmic evolution. Different galaxies, different star systems, different planets. Each represents a slightly different solution to the basic equations of physics and chemistry. Life, in its infinite creativity, explores these possibilities. And we, observing from our particular vantage point as conscious animals made of specialized cells, can appreciate the whole magnificent tapestry. Rest now in this diversity, in this richness of forms and functions, in this evidence that existence is creative, exploratory, generative. You are one solution among many, one form among infinite possible forms, and yet you are here. You are real. You are conscious and aware and preparing for sleep. That is enough. More than enough. You need not be anything other than what you are. Your cells are doing their work without your conscious direction, maintaining you, renewing you, participating in the ancient patterns of life that have continued for billions of years and will continue for billions more. Now we must zoom out slightly from our exploration of specific cellular structures and consider an astonishing fact. There exist organisms that consist of only a single cell, yet that cell contains everything necessary for complete, independent life. These single-celled organisms, or unicellular organisms, are not simplified or incomplete, but are fully functional living entities, capable of sensing their environment, obtaining food, reproducing, and responding to threats. The most common single-celled organisms are bacteria, prokaryotic cells that lack membrane-bound organelles. Bacteria are typically much smaller than eukaryotic cells, often just 1 or 2 micrometers long and have a relatively simple structure. A typical bacterium has a cell membrane surrounded by a cell wall, cytoplasm containing ribosomes, and a single circular chromosome of DNA, and sometimes additional features like flagella for movement or pili for attachment. Despite their simplicity compared to eukaryotic cells, bacteria are extraordinarily successful. They inhabit virtually every environment on Earth, from deep ocean trenches to high atmosphere, from frozen Antarctica to boiling hot springs, from the driest deserts to the saltiest lakes. They live on your skin, in your gut, in your mouth, everywhere. Most are harmless or beneficial. Only a small fraction cause disease. In fact, your body contains approximately as many bacterial cells as human cells, though they are so much smaller that they contribute only a few pounds to your total weight. Bacteria are metabolically diverse, far more so than eukaryotes. Some bacteria conduct photosynthesis, though not with chloroplasts, but with photosynthetic pigments embedded directly in their cell membranes. Others extract energy from chemical reactions, oxidizing iron, sulfur, or even hydrogen gas. Some bacteria can fix nitrogen from the atmosphere, converting it into ammonia that plants can use, making these bacteria essential to global nitrogen cycles. Others decompose organic matter, returning nutrients to ecosystems. The simplicity of bacteria should not be mistaken for primitiveness. Bacteria have been evolving for over 3 billion years, and they are exquisitely adapted to their environments. A bacterium can reproduce in as little as 20 minutes under optimal conditions, meaning that evolution can test new genetic variations very rapidly. This rapid reproduction and the enormous population sizes of bacteria mean that they evolve much faster than larger organisms, allowing them to adapt quickly to new antibiotics, new environmental conditions, new ecological niches. Archaea are another domain of prokaryotic single-celled organisms, superficially similar to bacteria, but genetically and biochemically distinct. Many archaea live in extreme environments, halophiles in saturated salt solutions, thermophiles in water near boiling, acidophiles in extremely acidic conditions, methanogens in oxygen-free environments where they produce methane. These extremophiles, as they are called, demonstrate that life's range is far greater than we once thought, that living cells can function under conditions that would quickly destroy most organisms. But single-celled eukaryotes, organisms whose cells have nuclei and membrane-bound organelles, are perhaps even more fascinating because they show us what a single cell can achieve when it has the full eukaryotic toolkit. These organisms are collectively called protists, though this is more a term of convenience than a taxonomic group. Protists are incredibly diverse, including organisms as different as amoebas, paramecium, euglena, diatoms, and many others. An amoeba is a single cell that moves by extending pseudopodia, temporary projections of its cell membrane and cytoplasm. These pseudopodia flow around food particles, engulfing them through phagocytosis, and can also be used for movement. The amoeba essentially flowing across surfaces. Despite being a single cell, an amoeba can sense chemical gradients in its environment and move toward food or away from harmful substances. A paramecium is covered with thousands of cilia that beat in coordinated waves, propelling the cell through water with surprising speed and maneuverability. A paramecium has multiple nuclei, a large macronucleus that controls everyday cellular functions, and one or more small micronuclei involved in sexual reproduction. It has a specialized region called a cytostome, or cell mouth, where food particles are ingested, and an anal pore, where waste is expelled. It has contractile vacuoles that pump excess water out of the cell, preventing the cell from bursting in fresh water. In short, a paramecium has organized its single cell to perform all the functions that in multicellular organisms might be distributed among many specialized cells and tissues. Euglena is particularly interesting because it blurs the line between plant and animal. A euglena has chloroplasts and can conduct photosynthesis when light is available, but it can also absorb organic molecules from its environment when kept in darkness, making it a mixotroph, capable of both autotrophic and heterotrophic nutrition. It has a flagellum for movement and an eye spot that detects light, allowing it to swim toward optimal light levels for photosynthesis. A single euglena cell thus combines features we typically associate with plant cells and features we associate with animal cells, reminding us that these categories are human constructs, convenient but not absolute. Single-celled organisms demonstrate that cellular complexity can be remarkable, even in the absence of multicellularity. A paramecium or euglena is far more complex than any individual cell in your body because it must perform all life functions within a single cell without the division of labor that multicellularity allows. Your nerve cells can specialize in signal transmission because other cells handle digestion, circulation, waste removal. A paramecium must handle all these functions itself, and it does so with impressive efficiency. The existence of single-celled organisms raises profound questions about what it means to be an individual, to be an organism. Is a single cell an individual? Clearly, yes, in the case of bacteria or paramecia. But what about the cells in your body? They are alive. They have all the characteristics of life. Yet we do not typically think of them as individuals, but as parts of you, the larger organism. Yet from the cell's perspective, if cells have perspectives, is there any difference? Each cell lives its life, performs its functions, responds to its environment, reproduces through division. Perhaps the distinction between single-celled and multicellular organisms is less sharp than we imagine. Perhaps you are better thought of as a colony of cells that have agreed to cooperate so extensively that they have created an emergent property, a consciousness that identifies as a single entity. From this perspective, the boundary between self and other, between one organism and many, becomes fuzzy, conceptual, a matter of perspective and definition rather than an absolute fact. When we think about cosmic parallels, single-celled organisms remind us that organization, complexity, and function do not require large size. A single cell, properly organized, can be a complete world. Similarly, a single star, properly situated, can be a complete system with planets orbiting, life potentially emerging, complexity arising. Size is not synonymous with significance. A bacterium is as perfect an example of adaptation and evolution as an elephant. A single star system with life would be as cosmically important as an entire galaxy. The universe creates complexity at all scales, from subatomic particles to superclusters of galaxies. And at every scale, we find organization, pattern, structure. Single-celled organisms occupy the microscopic scale, yet they are complete unto themselves, lacking nothing essential, demonstrating that wholeness is not a matter of size, but of integration, of all necessary functions being present and coordinated. As you drift toward sleep, consider that you began life as a single cell, a fertilized egg. That cell contained all the information necessary to build you, and through countless divisions, through differentiation into hundreds of cell types, through years of growth and development, that single cell became trillions of cells working together as you. Yet in a sense, you are still that single cell, grown large and complex, but fundamentally the same individual, the same continuous process that began at conception and will continue until death. There is poetry in this, in the recognition that single cells can be complete worlds, that complexity can scale from the microscopic to the macroscopic, that organization can arise at any level where the laws of chemistry and physics permit. You are made of cells, made of molecules, made of atoms, made of subatomic particles, made ultimately of quantum fields fluctuating in the void. At every level of description, you are complete. You are whole. You are a valid way for the universe to organize itself. Let this understanding dissolve any feeling of being incomplete, insufficient, not enough. Like a bacterium in its drop of water, you are a complete world, lacking nothing essential, perfectly adequate for your own existence. You need not be larger, more complex, different than you are. You are what you are. A pattern of organization that emerged from simpler patterns and will eventually dissolve back into simpler patterns. And for now, for this moment, you are conscious, aware, alive, and that is everything. We touched briefly on centrioles earlier, but they deserve a closer look, particularly in the context of cell division, one of the most fundamental and fascinating processes in cellular biology. Cell division is how one cell becomes two, how organisms grow, how injuries heal, how life continues across generations. It is a process so precise, so reliable, that errors are rare. Yet when errors do occur, the consequences can be severe, including cancer. Centrioles, as we discussed, are cylindrical structures made of microtubules, typically found in pairs oriented at right angles to each other. In animal cells, this pair of centrioles, along with surrounding material called the pericentriolar material, forms the centrosome, the primary microtubule organizing center. Before a cell divides, the centrosome duplicates, so that the cell briefly has two centrosomes, each with its pair of centrioles. As the cell prepares for division, these two centrosomes begin to move apart, migrating to opposite sides of the nucleus. Microtubules grow from each centrosome, reaching into the cell, some microtubules growing long enough to interact with microtubules from the opposite centrosome. This forms the mitotic spindle, a beautiful structure resembling a three-dimensional football or rugby ball, with the two centrosomes at opposite poles and microtubules radiating from them. Meanwhile, inside the nucleus, the chromatin has been condensing into distinct chromosomes. Each chromosome at this point consists of two identical sister chromatids, the result of DNA replication that occurred earlier in the cell cycle. These sister chromatids are held together at a region called the centromere. As the nuclear envelope breaks down, a major event that marks the transition from prophase to metaphase, the chromosomes become accessible to the mitotic spindle. Some microtubules from the mitotic spindle attach to structures called kinetochores, which form at the centromeres of chromosomes. Each sister chromatid has its own kinetochore, and microtubules from opposite poles of the spindle attach to these kinetochores, creating a bipolar attachment. The chromosomes are then moved to the cell's equator, forming the metaphase plate, an imaginary plane equidistant from the two spindle poles. All the chromosomes line up along this plane, and the cell pauses here in metaphase, while checkpoint mechanisms verify that every chromosome is properly attached to the spindle. This checkpoint is crucial. If even one chromosome is not properly attached, if even one kinetochore lacks proper bipolar attachment, the cell will not proceed to the next stage. This pause allows time for corrections, for improper attachments to be dissolved and proper attachments form. Only when all chromosomes are correctly attached, with each sister chromatid connected to opposite poles of the spindle, does the cell receive the signal to proceed. When the signal comes, the proteins holding sister chromatids together are suddenly cleaved, and the sister chromatids separate, becoming individual chromosomes. The microtubules attached to the kinetochores begin to shorten, pulling the now separated chromosomes toward opposite poles of the cell. Simultaneously, the spindle itself elongates, pushing the poles farther apart. The result is that two identical sets of chromosomes move to opposite ends of the cell, each set containing one copy of each chromosome. As the chromosomes reach the poles, the cell begins to physically divide through a process called cytokinesis. In animal cells, a ring of actin filaments forms at the cell's equator, perpendicular to the spindle axis. This contractile ring, working with myosin motor proteins, tightens like a drawstring, creating a cleavage furrow that deepens until the cell is pinched into two. In plant cells, Cytokinesis occurs differently because the cell wall prevents the cell from being pinched. Instead, a new cell wall, called the cell plate, forms between the two daughter nuclei, growing outward until it fuses with the existing cell wall, dividing the cell into two. The result of mitosis and cytokinesis is two daughter cells, each with a complete set of chromosomes, identical to the parent cell, each with its own nucleus, its own organelles, its own cell membrane. These daughter cells may then grow, carrying out normal cellular functions, until perhaps they too divide, continuing the cycle. There is something almost miraculous about the precision of mitosis. Consider that a human cell has 46 chromosomes, and each must be duplicated, condensed, attached to the spindle, aligned, separated, and distributed to daughter cells without error. The probability of error at any step is non-zero. Yet the entire process usually proceeds flawlessly, completing in about an hour, ensuring that genetic information is faithfully transmitted from parent to daughter cells. When errors do occur, the consequences vary. Sometimes a chromosome fails to separate properly, and one daughter cell receives two copies, while the other receives none. This is called non-disjunction, and in most cases, the affected cells die because they cannot function without the correct number of chromosomes. But sometimes such cells survive, and if they continue dividing, they can give rise to tissues with abnormal chromosome numbers, potentially leading to cancer or developmental abnormalities. The elegance of cell division, the choreography of chromosomes and spindle microtubules, the precision of timing and checkpoint controls, all demonstrate the sophistication of cellular machinery. This process has been refined over billions of years of evolution, with countless variations tested and the most reliable mechanisms preserved and passed down. What we observe in any dividing cell today is the culmination of this long evolutionary process, a solution to the problem of how to reliably copy and distribute genetic information. In the context of our cosmic comparisons, cell division might remind us of stellar binary systems, where one star eventually fissions, or more accurately, of molecular clouds, fragmenting to form multiple stars. When a large molecular cloud collapses under its own gravity, it often fragments into multiple clumps, each of which may collapse further to form a star. This is not exactly like cell division, which is a controlled, precise process, but it shares the theme of one becoming two, of division creating multiplicity. More broadly, Cell division exemplifies a fundamental principle. Complex systems often reproduce by copying themselves. The most efficient way to create another complex system is not to build it from scratch, but to copy an existing one. Cells divide, copying themselves. Organisms reproduce, passing on genetic information that allows offspring to develop. Stars form from collapsing gas clouds that fragment. Galaxies may merge, or gas within galaxies may fragment to form new stellar populations. At every level, complexity begets complexity through processes of replication and division. As you lie here, cells throughout your body are dividing. In your bone marrow, stem cells are dividing to produce new blood cells, replacing the millions of red blood cells that die every second. In your digestive tract, epithelial cells are dividing to replace cells that are constantly sloughed off. In your skin, cells are dividing to repair minor damage, to maintain the barrier between you and the environment. This constant cellular division is what maintains you, what allows you to persist, even though your individual cells are constantly dying and being replaced. The fact that you are the same person you were 10 years ago, even though most of your cells have been replaced, raises deep questions about identity and continuity. What makes you you if your physical substance is constantly changing? Perhaps it is the pattern, the organization, the information encoded in your cells, rather than the specific atoms that constitute you. Like a river that remains recognizably itself, even though the water is constantly flowing and changing, you remain yourself through constant cellular turnover. Let this understanding help you release attachment to fixed identity, to the illusion of permanence. You are a process, not a thing, a pattern that maintains itself through change, a dance of molecules that somehow gives rise to consciousness and experience. And that is beautiful, more beautiful perhaps than if you were fixed and unchanging. Change is not something happening to you, but something you are. Fundamentally, at every level, from cellular division to psychological growth. As sleep approaches, feel yourself dissolving slightly, your grip on waking identity loosening, your sense of being a fixed, separate self softening into something more fluid, more process-like. This is not something to fear, but something to embrace. A small taste of the constant flux that is actually always happening, usually hidden beneath the illusion of stability. Your cells divide, your molecules turn over, your thoughts flow and change. And yet something persists, some organizing principle, some pattern that is recognizably you. Trust in that pattern, in that persistence, through change, in that continuity that does not require fixity. We have mentioned ribosomes several times in our journey, but now we must give them the attention they deserve as perhaps the most abundant molecular machines in cells, the structures responsible for translating genetic information into functional proteins. Ribosomes are found in all cells, both prokaryotic and eukaryotic, because all cells must synthesize proteins, and ribosomes are the only structures capable of performing this essential task. A ribosome is not an organelle in the traditional sense, because it is not enclosed by a membrane. Instead, it is a complex molecular machine made of ribosomal RNA, or rRNA, and many different proteins. Each ribosome consists of two subunits, a large subunit and a small subunit, that come together when protein synthesis begins and separate when it ends. In eukaryotic cells, the small subunit contains one rRNA molecule and approximately 33 different proteins, while the large subunit contains 3 rRNA molecules and about 49 different proteins. Together, they form a structure about 20 to 30 nanometers in diameter, large enough to be visible in electron micrographs as small dots. The process of protein synthesis, called translation, is one of the central processes of molecular biology. The step where information encoded in genes is converted into functional molecules. It begins when a messenger RNA molecule, or mRNA, which carries the genetic instructions copied from DNA in the nucleus, binds to a small ribosomal subunit. The mRNA contains the information for one protein, encoded as a sequence of 3-letter codons, each codon specifying one amino acid. The small subunit positions itself at the start of the coding sequence on the mRNA, and then the large subunit joins, creating a complete ribosome with the mRNA threaded between the two subunits. The ribosome has 3 binding sites for transfer RNA molecules, or tRNAs. Each tRNA carries a specific amino acid and has an anticodon, a 3-nucleotide sequence that pairs with a complementary codon on the mRNA. As the ribosome moves along the mRNA, reading each codon in sequence, appropriate tRNAs bind bringing their amino acids into position. The ribosome catalyzes the formation of peptide bonds between adjacent amino acids, linking them into a growing chain. When the ribosome reaches a stop codon, a codon that does not code for any amino acid, protein synthesis terminates. The completed protein is released, and the ribosome subunits separate. This description makes translation sound simple and linear, but it is actually extraordinarily complex and precise. The ribosome must discriminate among dozens of different tRNAs, selecting only the one with the correct anticodon for each codon. It must catalyze peptide bond formation with high fidelity, ensuring that amino acids are joined in precisely the right order. It must move along the mRNA exactly 3 nucleotides at a time, maintaining the correct reading frame. Because a shift of even 1 nucleotide would change all subsequent codons and produce a completely different, almost certainly non-functional, protein. The ribosome accomplishes this through its intricate, 3-dimensional structure with precisely positioned chemical groups that bind to tRNAs, mRNA, and the growing protein chain. Much of the catalytic activity is actually performed by the rRNA, not by proteins, making the ribosome a ribozyme, a catalyst made of RNA rather than protein. This suggests that ribosomes may have originated very early in the history of life, perhaps in an RNA world, before protein enzymes evolved, when RNA molecules performed most of the catalytic functions in primitive cells. Cells contain enormous numbers of ribosomes. A rapidly growing bacterial cell might contain 20,000 ribosomes, and a eukaryotic cell can have millions. These ribosomes are working constantly, synthesizing the thousands of different proteins the cell needs. Enzymes to catalyze metabolic reactions, structural proteins to build cytoskeleton and organelles, transport proteins to move molecules across membranes, regulatory proteins to control gene expression, receptors to sense signals, motor proteins to generate movement, and countless others. Ribosomes can be found free in the cytoplasm, where they synthesize proteins that will function in the cytoplasm, or in organelles, or attached to the endoplasmic reticulum, where they synthesize proteins destined for secretion or for insertion into membranes. Some ribosomes are found in mitochondria and chloroplasts, which have their own ribosomes, similar to bacterial ribosomes. Another piece of evidence supporting the endosymbiotic origin of these organelles. The process of translation consumes significant energy. Each peptide bond formed requires the energy from several molecules of GTP, a molecule similar to ATP. Each tRNA must be charged with its amino acid, a process that requires ATP. Given that proteins can contain hundreds or thousands of amino acids, and that cells must synthesize thousands of different proteins, the energy cost of protein synthesis is substantial, probably accounting for a large fraction of the cell's total energy budget. Yet this investment is necessary because proteins are what actually do most of the work in cells. DNA stores information, but DNA itself is chemically inert, does not catalyze reactions, does not form structures, does not move or transport or respond. Proteins, with their diverse shapes and chemical properties, perform all these functions. The flow of information from DNA to RNA to protein, called the central dogma of molecular biology, is the fundamental process by which genetic information becomes biological function. When we search for cosmic parallels, ribosomes and protein synthesis remind us that information and its physical manifestation are two different things. In galaxies, the distribution of dark matter provides information about gravitational potential, determining where gas will accumulate, where stars will form, how the galaxy will evolve. But this information must be translated into actual structures, stars, planets, molecular clouds. The information exists as gravitational fields, but the manifestation exists as matter and energy, organized in specific ways. Similarly, genetic information exists as sequences of nucleotides in DNA, but the manifestation of this information exists as proteins folded into specific three-dimensional shapes, performing specific functions. The ribosome is the translator, the machine that converts information into matter, abstraction into reality, code into function. The existence of this translation process, this conversion of information into physical form, is profound. It means that life is not just matter, but also information. Not just stuff, but also pattern. Not just molecules, but also meaning. The sequence of nucleotides in DNA means something. It codes for specific amino acid sequences, which fold into specific protein shapes, which perform specific functions. This semantic dimension of life This presence of meaning and information distinguishes living systems from non-living matter. As you drift toward sleep, your ribosomes continue their constant work, synthesizing the proteins you will need tomorrow, replacing proteins that have been damaged or degraded, building the molecules that will repair tissues, fight infections, transmit neural signals, contract muscles. This work continues without your awareness, without your intention, guided only by the information in your genes and the regulatory signals from your cell's environment. There is profound trust required to live. Trust that your ribosomes will continue synthesizing proteins, that your mitochondria will continue generating ATP, that your heart will continue beating, that your lungs will continue breathing, all without your conscious control. You cannot choose to keep your cells alive. You can only allow them to do what they do, to follow the patterns encoded in their structure and information, to live as they have lived for billions of years. Let this recognition of automatic function, of processes that continue without effort or attention, help you release conscious control. Help you trust in the underlying mechanisms that sustain life, that require no conscious direction. You do not need to manage every aspect of your existence. Most of it manages itself, has managed itself since before you were born, will manage itself after you fall asleep. Your task is simply to allow this, to not interfere, to trust in the wisdom of 4 billion years of evolution, to rest in the knowledge that life knows how to live. We have journeyed together through the landscape of the cell, from the phospholipid bilayer of the cell membrane to the genetic information in the nucleus, from the energy-generating mitochondria to the protein-synthesizing ribosomes, from the processing centers of the endoplasmic reticulum and Golgi apparatus to the recycling centers of lysosomes and peroxisomes, from the structural framework of the cytoskeleton to the specializations of plant and animal cells, from the complete worlds of single-celled organisms to the precision of cell division. It has been a long journey, measured not in distance, but in complexity, not in space, but in understanding. And throughout this journey, we have occasionally lifted our gaze from the microscopic to the cosmic, noting parallels between cellular structures and galactic structures, between the organization of cells and the organization of galaxies. These parallels are not coincidental, but neither are they evidence of some mystical connection between cells and galaxies. Rather, they reflect fundamental principles of organization that apply at multiple scales. The efficiency of boundaries, the importance of energy gradients, the utility of networks, the necessity of information storage and processing, the elegance of recycling and renewal. Nature, it seems, discovers certain solutions again and again, applying them at different scales with different materials. The same principles that govern the structure of cell membranes also govern the boundaries of galaxies. The same logic that makes networks efficient for material transport in cells also makes networks efficient for matter distribution in cosmic structures. The same necessity for energy generation that drives mitochondrial evolution also drives stellar evolution. The same requirement for information storage that leads to nuclei in cells leads to concentrated mass and organization in galactic cores. These patterns suggest that the universe is not chaotic, but ordered, not random, but structured, not arbitrary, but governed by comprehensible principles. This is a deeply comforting thought for a mind-seeking rest. The universe makes sense. Reality follows rules. Patterns exist and persist. From the smallest cell to the largest galaxy, from the briefest moment to the longest age, there is organization, there is logic, there is something we can recognize as reason. You lying here at the edge of sleep are part of this patterned universe. Your cells follow the same principles we have discussed tonight. Your body is organized according to rules that apply equally to bacteria and to galaxies. You are not separate from nature, but are nature, not outside the patterns, but woven into them. Not an observer only, but also the observed. Not just conscious, but consciousness itself. The universe becoming aware of its own organization. The journey inward into the cell is also a journey inward into yourself, because you are made of cells constituted by cellular processes, sustained by cellular metabolism. When we describe a mitochondrion generating ATP, we are describing something happening inside you right now. When we describe a ribosome synthesizing protein, we are describing the mechanism by which you maintain and renew yourself. When we describe a nucleus storing genetic information, we are describing the repository of the instructions that make you uniquely you. And the journey outward to the galaxies is also a journey outward to your context, your place in the cosmos. The atoms in your body were forged in stars, scattered through space by supernovae. Eventually, coalescing into the solar system and Earth. You are literally made of stardust, literally composed of material that once floated between galaxies, that once formed other stars, other planets, perhaps other living beings. Your connection to the cosmos is not metaphorical, but physical, material, absolute. Between the cellular and the galactic, you exist as a human being made of trillions of cells organized into tissues and organs and systems, living on a planet orbiting a star in a galaxy containing hundreds of billions of other stars. This is your scale, your size, your natural level of organization. But knowing the scales above and below, understanding the patterns that connect them, enriches your experience of your own level, helps you see yourself as part of a continuum rather than as an isolated entity. As sleep finally approaches, as consciousness begins to fade, as the boundary between waking and dreaming blurs, remember that what is happening to you now, this transition into sleep, is also a pattern that repeats, that follows rules, that is part of the natural order. Every organism that has a nervous system experiences something analogous to sleep. Every organism that has cells experiences circadian rhythms, daily cycles of activity and rest, driven by ancient molecular clocks. Your sleep is not a departure from existence, but a different mode of it. Not an absence, but a presence of a different kind. While you sleep, your cells continue their work. Your brain consolidates memories. Your body repairs damage. Your immune system patrols for threats. Life continues. Patterns persist. Organization is maintained. You do not need to remain conscious to remain alive, to remain yourself, to remain part of the great pattern. Let yourself sink into sleep as a cell sinks into the cytoplasm, surrounded by supporting structure, part of a larger whole, individual yet connected. Let your consciousness drift like a molecule in Brownian motion, random yet bounded, free yet part of a system. Let your thoughts slow like cellular metabolism in dormancy, still present but quiet, still active but restful. The cell membrane separates inside from outside, but permits exchange. Your skin separates self from world, but permits interaction. Your consciousness separates awareness from environment, but permits experience. These boundaries are necessary, functional, life-enabling, yet they are not walls but membranes, not barriers but interfaces, not separations but gradients across which things flow. As you cross the boundary into sleep, remember that you are crossing a membrane, moving from one state to another, but remaining yourself, transitioning from waking to dreaming, but remaining continuous, changing modes, but not changing essence. The patterns that organize you remain, the principles that sustain you persist. The structures that constitute you continue their work. You are a cell in the body of humanity, which is a species on the surface of Earth, which is a planet orbiting a star, which is a point of light in a galaxy, which is a collection of matter and energy in the cosmos. At every level, you are part of something larger, and at every level, you contain something smaller. You are both container and contained, both part and whole, both individual and collective. This nested organization, this hierarchy of scales, this pattern of patterns, is perhaps the most fundamental truth of existence. Everything is made of smaller things and is part of larger things. Everything is organized and organizes. Everything is process and persistence, change and continuity, flow and form. As your eyes close, as your breathing deepens, as your muscles relax, as your thoughts scatter and dissolve, trust that the patterns will hold, that the organization will persist, that tomorrow you will wake still yourself, still organized, still functional. The cell membranes will still separate inside from outside. The nuclei will still store genetic information. The mitochondria will still generate energy. The ribosomes will still synthesize proteins. And in the galaxies, light years away, stars will still burn. Planets will still orbit. Matter will still organize into patterns. The universe will continue being itself, following its rules, manifesting its patterns, creating complexity from simplicity, information from matter, consciousness from chemistry. You are part of this, essential to it, necessary to the universe's ongoing exploration of possibility. Your existence matters. Your consciousness counts. Your experience is the universe experiencing itself. When you wake tomorrow, you will continue the ancient pattern of life, of awareness, of participation in cosmic becoming. But for now, sleep. Rest in the patterns, trust in the organization, surrender to the processes that know how to sustain life without conscious direction. Let the cell membrane of consciousness become permeable. Let thoughts drift in and out. Let awareness fade like cytoplasm at the edge of a cell, not disappearing, but diffusing, not ending, but blending into something larger. Sleep well, fellow traveler through scales of existence, fellow pattern in the cosmic organization, fellow cell in the body of reality. The patterns hold you, support you, sustain you. The universe is your membrane, your cytoplasm, your nucleus. You are safe within its structures, protected by its organization, maintained by its eternal processes. Good night. Rest. Dream of cells and galaxies, of membranes and boundaries, of inside and outside, of patterns repeating at every scale. Dream of yourself as you are. Organized complexity, conscious matter, the universe awakening to itself. And know that when consciousness returns, you will be renewed, refreshed, ready to live another day as a miracle of cellular organization. He is a human being aware of the patterns that constitute existence, as a point of awareness in the vast, patterned, meaningful cosmos. The journey from cell nucleus to galactic center continues, but for now, you rest at the center of your own experience, at peace, at home, at one with the patterns that make you possible. Sleep now. The cells know what to do. The patterns will hold. Everything continues, including you, even in sleep, even in dreams, even in the quiet darkness where consciousness dissolves back into the mystery from which it arose. Rest now. Sleep. The universe continues its work, and so do you, even in sleeping, even in letting go, even in surrender to the patterns that sustain all things at all scales, from the smallest cell to the largest galaxy, from this moment to eternity. Good night.